All right, I think we're good to go. So good afternoon to everyone. My name is Tuli Reister, and I am the project manager of the IAS Corporate Partnership Program. And it is my great pleasure to open this Industry Towards an HIV Cure Collaboration Group workshop on non-invasive measurement of HIV reservoirs. As a reminder, let me go over a couple of housekeeping rules for the workshop. So this workshop will be recorded, and the recording will be made available to the participants and on the IAS website as part of the educational materials in the coming weeks. By staying in this meeting, you are essentially consenting to being recorded during the workshop. We encourage you to send questions to our presenters at any time through the chat function at the bottom of your screen and during the Q&A session by raising your hand. The button is also at the bottom of your screen. And please do remember to focus your questions on today's workshop. Before we move forward, I would like to hand over to the Towards an HIV Cure Industry Collaboration Group co-chair, Bonnie Howell, for a brief introduction of today's workshop. Thank you.
Great. Thank you, Tuli, and welcome everyone. So good morning, good afternoon. My name is Bonnie Howell. I am Vice President of Pharmacology and the Global HIV Cure Lead at Merck & Company in the US. This workshop will bring together industry representatives and industry collaboration group, as well as researchers and clinicians that will discuss the existing assays, as well as innovative non-invasive technologies for the measurement of the HIV reservoir. And so we know that there have been several approaches that have been developed to date to measure the HIV reservoir. And so the workshop will focus on the much needed non-invasive techniques and surrogate markers that we could use to detect and quantify the reservoir in a range of cells and tissues, so extending even beyond blood. And we hope that these reservoir assays will be able to be used in a variety of settings for which HIV cure research is conducted. We feel that the identification of the markers could accelerate the development and the assessment of HIV cure interventions by providing mechanistic insight, possibly leading to the development of new therapeutic approaches, as well as providing an alternative to or a complement to the analytical treatment interruption and viral load measurements commonly used in cure research. And so I'd like to welcome all of the participants and our guest presenters, and I will now hand it over to my co-chair, Tim, who will also do a brief welcome. Tim, over to you.
Thanks, Bonnie. Good morning and good afternoon, good evening, everybody. I'm Tim Henrich, Associate Professor of Medicine at UCSF. And as Bonnie said, it's a real pleasure to be here to explore, discuss opportunities, challenges, et cetera, when it comes to measurement of HIV reservoirs. And I think this is really an important topic that I know everyone here on the call is intimately familiar and has discussed in certain forms. And but in the first part of this workshop, we will have 5 presentations followed by a Q&A session after those presentations. So please hold questions until after. You can certainly put things in the chat. The second part will include 4 short presentations, these flash talks, followed by group discussion in 2 breakout rooms, which I think will really be the meat of participating. So we're hoping that those who are on the call, not just talking, will really be able to participate and be heard. And this is going to be very important for the IAS and as we go forward thinking about non-invasive reservoir measurements. So we do have a packed agenda. So we please ask all speakers not to go over their associated time slots. Bonnie and I will be the enforcers. And so we will remind folks if they are going over. So please keep it into the time so we can get to the discussion towards the end as well. And with that, let's dive right in. I'm going to hand over to Dr. Nicolas Chaumont, who also needs no introduction to this group, Associate Professor in the Department of Microbiology, Infection, and Immunology at the University of Montreal. We'll be speaking on HIV reservoir measurements today, and we'll kick off with an overview. Nicolas.
Thank you very much, Tim. It's a pleasure to be here. Thank you for the invitation. So it's a huge task to try to summarize everything we know about HIV reservoir measures in 10 minutes. So don't expect that because it's just impossible. So I decided to select a few aspects of these reservoir measures that we've studied, not only me, but many people in the field during the past 20 years and try to convey what I think is the most important message is that the best assay to be used for your study really depends on the question you're asking. So first, maybe I would like to remind you why we need to measure the reservoir. Well, actually, we have good measures of active viral replication before antiretroviral therapy. So that's plasma viral load, basically. And you can also monitor HIV rebound during antiretroviral therapy interruption. And you can use plasma viral load to monitor rebound. So when people are not on ART, basically the answer is quite easy. You use plasma viral load and you can monitor either the progression of the disease or the viral rebound. But the big question is, what do we do in people who are fully suppressed on ART? What do we need to, what should we measure? And as you know, there are many assays that have been developed again during the past 20 or 30 years. So the question here again is, the assay you will pick really depends on the question you're asking. So if you're trying to measure a decay in the size of the reservoir during fully suppressive therapy, the assay you will use might not be the same that the one you would pick if you want to predict viral load rebound after treatment interruption. And I will show you some examples later. So the big challenges when we want to measure the reservoir during ART is that the cells in which HIV persists are rare. They are in many different deep tissues, and I think we'll hear from Claire Deléage and Zaza and Lovu about this. And obviously, again, choosing the good assay requires to ask the right question. And that seems a little bit trivial, but it's not. Because we need to exactly define what would be targeted by a cure strategy to be able to choose the best suited assay for your question. So as you know, there are many things we can measure. This is a complicated slide, but basically what it says is that in latently infected cells, obviously there is HIV DNA, and this DNA is most of the time integrated into the chromatin of the cell. And when you stimulate the cell with your favorite stimuli, that could be CD3, CD28, or PMA and ionomycin or others, this DNA will be maintained in the cell, obviously, but other things will appear, such as cell-associated HIV RNA, possibly viral proteins, and finally viral particles. So during ART, HIV DNA can be detected in blood and tissues in most people, but viral RNA and viral proteins are rare, but they can be induced if you stimulate the cell, again, with either through the TCR or through either pathways. The viral load growth assay was the gold standard for many years, and we've all used this assay that was originally developed by Bob Silicano, and then many people in the field, including Nancy Archin and Dave Margolis, have actually used this assay and confirmed the half-life of the reservoir using what was the gold standard. And I say was the gold standard, you might think it's still the gold standard because this is actually the only assay that can measure the frequency of cells harboring replication-competent HIV. It's a complicated assay, quite annoying. So those who have run it in a lab, you know that. You need to isolate CD4 cells, plate them in limiting dilution, then you activate them, you add feeder cells, and then you measure P24 antigen after 2 or 3 weeks of culture. So that's quite complicated, but that gives you really the number of the frequency of cells that have this famous replication-competent virus, which is really the one you'd like to measure in cure studies. The problem is that the QVOA, it's not a problem, but the first paper, and this is really an iconic figure from the Erickson paper, and many different groups actually contributed to this work, in which they reported that there is a 2-log difference between the level of HIV DNA and the frequency of cells measuring harboring replication-competent HIV. So this 100-fold difference was somewhat unexpected, and the big question was, why do we see this 100-fold difference? And the answer came from many different studies, again, pioneered by Yachi O and Bob Silicano, who actually found that many of the proviruses in people on suppressive therapy are actually defective. So they have large internal deletion or other defects. And this DNA, even though they are integrated in the chromatin of the host cell, they cannot produce a fully competent viral particle. And this is the reason why DNA measures overestimate the size of the real viral reservoir. And there are many assays that have actually been developed, some by Sarah Palmer, who developed this FLIPS assay, and other, again, Yashiho, but also Matthias Lichtenfeld, and also Michel Nussenzweig have developed their own assay to measure this, or to sequence this near full genome. Near full HIV genome. This is an example of a work in my lab in which we worked on an autopsy study, and we actually collected many different tissues from this person and used a modified version of the FLIP assay from Sarah. And we found also that this is true in blood, but also in tissue. Most of the HIV DNA molecules persisting in people on suppressive therapy are defective. I don't know if you can see the little red sign on the right, but this red sign actually mark the intact genomes. And you can see that over 300, we sequenced over 300 genomes here, and I believe only 5 were genetically intact. So this is quite complicated to perform near full genome sequencing. So alternative assays have been developed, such as the intact profile DNA assay, or IPDA, which has been developed by the group of Bob Silicano again. This is a ddPCR, or a digital drop PCR based assay, a quantitative PCR method that amplifies frequently deleted regions in defective genomes, such as the packaging signal or the rev responsive element. And it also detects hypermutation. So I will not go into the detail of this assay, but the beauty of this is that because it's a PCR based assay, it's relatively easy to run. It's not that expensive, probably less expensive than near full genome sequencing, and can give you an idea of the frequency of supposedly genetically intact HIV genome in a given sample. And you can actually use it not only in blood, but also in tissues. The problem with IPDA, first, the prediction process is not perfectly, is not perfect. So you do not always catch only intact genomes with IPDA, or classify intact genomes with IPDA. And the other problem is that it's actually quite difficult to develop an IPDA that would work on every HIV clade. So just to summarize what I just told you, HIV DNA measures largely overestimate the real size of the reservoir, and QVUA does not capture all cells with genetically intact genomes. So all measures are basically imperfect. We've learned quite recently from Matthias Lichtenfeld that over time, the proportion of HIV genomes with active transcriptions diminishes during ART, and the remaining intact genomes concentrate in non-genic regions. And the consequence of this is that you would like, of course, to assess the genetic integrity of HIV genomes, which is important, but their inducibility, so basically their capacity to produce viral particles, might also be extremely important and might actually be reduced with time on antiretroviral therapy. And there are several reservoir induction assays that have been developed. We developed TILDA probably 5, 6, 7 years ago, I don't remember, HIV flow, which measures viral protein. But there are other assays that measure actually both RNA and P24. These are flow-based assays. John Mellors developed another assay in which he measured the frequency of cells producing viral particles. And VOA, the viral outburst assay, is also an induction assay that measures replication competent HIV. So I only have 2 slides left. I think this is probably one of the most important message I'd like to convey. How to choose your assay. I mean, there are different questions you need to ask yourself. First, what is your question? And that is the most important question, actually, you should ask yourself. The assay you will pick will not be the same if you try to monitor viral rebound, if you try to demonstrate that someone has been cured, or if you want to test the efficacy of a shock and cure strategy. What is the HIV clade? There are some assays that do not work on clades that are non-B. How many cells are available? Obviously, this is something that sometimes you cannot really control, and some assays require lots of cells because they measure events that are extremely rare. How many samples do you need to run, and what is your budget? This is an obvious question, not really scientific, but important for many in the field. And finally, what equipment is available? If you need to perform IPDA, you need a digital droplet PCR machine. If you want to perform HIV flow, you need a flow cytometer. So you will circle into this question, and at the end of the day, you always need to finish with this question. What is your original question? What are you targeting? And then you can go and proceed with your measures. My last two slides. Conclusions. Cells in which HIV persists are rare. We need many cells. We need sensitive assay and reproducible assay. HIV persists in many deep tissues, so we need a blood measure that reflects this anatomical reservoir, and I believe that Mohamed Abdelmohsen will talk about this later. We need non-invasive methods, such as imaging, and Tim will talk about it. We need to ask the right question to select the best suited assay. Measuring the efficacy of a shock and kill strategy will require different measures than for a gene therapy approach. What do we target? All HIV genomes are not genetically intact, and all genetically intact genomes are not necessarily inducible. What is the most important? Again, it depends on the question. To predict rebound upon ATI, measuring the inducibility might actually be a good idea. And this is really my last slide with the things we should not say anymore. We should not say that this assay is the best or the gold standard. It always depends on the questions you're asking. We should not say that you should only use resting CD4 T cell to measure the reservoir. I think we now have pretty good evidence that HIV can also persist in cells that express these so-called activation markers. Only intact genomes matter. Probably not if you're looking at comorbidities and inflammation. Defective genomes also have the ability to produce viral RNA and proteins. In addition, and this is something I did not mention, our prediction of intactness may not always be accurate. If you measure 4 copies of XYZ and then 2 copies of XYZ, that's not necessarily a 50% reduction. It depends on the precision and the reproducibility of your measure. And finally, if you don't detect something, it doesn't mean it's not there. It really depends on how much you sample, and we should always provide limits of detection based on cell input. I'll stop there. Thank you, and ask question later. I'm sorry, I'm 1 minute late, I believe.
Nicola, thank you. That was fantastic that you did it. You overviewed the entire field in 11 minutes. Thank you. And from now, we will move on to our next speaker, who also needs no introduction, Dr. Jonathan Lee from Harvard and Brigham and Women's, who will present about predictors of viral rebound. John.
Okay, thanks a lot, Tim. Thanks for inviting me to talk about reservoir predictors of HIV rebound after treatment interruption. So I will very briefly kind of go over a couple slides on background. You guys, I think all know this already, that successive interventions aimed at achieving HIV remission eventually will need to be tested by its ability to show clinically meaningful results, either by decreasing the set point or delay in rebound. But of course, there are downsides of treatment interruption trials, including retroviral rebound syndrome, drug resistance, HIV transmission, and of course, it is very logistically challenging and burdensome for both the participants and for the investigators to do. And these downsides that I've mentioned have been seen in clinical trials, such as the one that I'm showing here. And so, given the issues and risks with treatment interruption, identification of biomarkers to predict time to HIV rebound is going to be important in developing and evaluating promising treatment strategies. So we just heard a fantastic talk by Nicola about the range of HIV reservoir measures out there. And since there's no one assay that can fully represent the entirety of the reservoir, most clinical trials these days are using the strategy of testing multiple assays to provide a more holistic view of the reservoir. So I've just listed 7 different trials from the ACE Clinical Trials Group, as well as internationally, and all of the reservoir measures. Just to give you a sense of what people are using out there. Most people are using cell-switching DNA, cell-switching RNA, and more recently, the IPDA, as Nicola has gone over. Although the entirety of the reservoir measures that people are using does differ depending on what study you're looking at. And then people are also looking at more investigative, additional kind of more niche-based assays, as seen on the right-hand side here. So what are the data behind some of these assays in predicting HIV rebound timing. So why don't we start off by looking at HIV DNA levels. There's a couple papers from on the left from the Spartex study from John Frater's group, James Williams, and on the right from an ANRS group, both of these suggesting that HIV DNA levels can predict the timing of viral rebound. And then we had published a study in the ACTG looking at some of the older ACTG studies, New York Central Issue 371, showing that a smaller expressed HIV reservoir can predict delayed viral rebound, especially cell-social RNA levels on the right and residual viremia by the single copy assay, or I'm saying cell-social RNA on the left, residual viremia by the single copy assay on the right-hand side, both with lower levels seen in individuals with more delayed viral rebound. And these reservoir measures were tested before treatment interruption. In addition, in a study of a therapeutic vaccine study in early treated individuals, this was done by Taiwo Chung and Tony Fauci's group, they also saw that amongst the biomarkers they looked at, the cell-social RNA levels was the most strongly predictive of the timing of viral rebound. But what we really have needed in the field, I think, is a systematic analysis of the reservoir predictors of HIV rebound in both early and chronic individuals, chronically treated individuals, and that's where ACTG 5345 has come in. This is a study that led by myself and also my co-chair, Davey Smith, and the aim is to determine the virologic, immunologic host predictors of the time to viral rebound. And we enrolled individuals who initiated ART during either chronic infection or during early infection. Individuals who are on an NNRTI-based regimen were switched to an integrase inhibitor-based regimen. They underwent a treatment interruption, had intensive follow-up, twice a week viral load monitoring, and then once their viral load reached 1,000 copies, then they reinitiate ART, and we follow them again on ART. And in terms of the overall viral rebound kinetics, the median time to viral rebound was about 3 weeks, 22 days. One participant in each group did not meet ART restart criteria for at least 24 weeks. When we compared the timing of viral rebound of these individuals in 5345 who are on modern ART regimens to individuals part of historic ACTG studies who are on really older PI-based regimens, we actually did not see a significant difference in the timing of viral rebound. So modern ART did not appear to delay viral rebound. And we did find a modest delay in time to rebound for early treated participants compared to chronic treated individuals. And in terms of the reservoir and other assays that were tested in 5345, for the pretreatment time points, for the virology assays, we looked at unspliced cell-social RNA, total cell-associated HIV DNA, the IPDA done by Elsevier, residual viremia by the ISCA, and then IUPMs by the differentiation quantitative viral outgrowth assay done by Southern Research. Antibody levels were measured by Mike Bush's group, T-cell phenotyping by Alan Lande, HIV-specific T-cell responses by Bish Makatange's group, and then soluble inflammatory markers by Mohammed Abdel Mohsen, who's actually going to be giving a talk right after me. So let's jump into some of the results of 5345. So what were some of the reservoir predictors of HIV rebound timing? So I initially talked about some of the older trials looking at cell-social RNA and DNA. And I would say, there is some very modest correlations here. The p-values were not, did not fully reach significance. I think some of that had to do with some of the limited sample size and participants in 5345. But all of these measures here across all of the different participants had some modest negative correlation with timing of viral rebound. But the strength of the correlations really does differ by the timing of ART initiation. In chronic treated individuals, actually the IPDA and the intact per-hour DNA level was the value that was most strongly associated with the timing of viral rebound, with higher levels of intact per-hour DNA associated with a shorter, faster timing of viral rebound. And then for early treated individual, it was actually the single copy assay or the ISCA that was most strongly associated. So we had just talked about, that there are HIV reservoir measures that can predict viral rebound. But what happens in a setting where you have extreme reservoir depletion without an HIV-specific immune response? So I'm going to point to a study that was actually led by Tim Henrich. this is a study of two individuals who both had hematologic malignancies and underwent hematologic stem cell transplantation. And both donors eventually achieved full donor chimerism. And when Tim looked at the HIV DNA levels, couldn't actually find any HIV DNA in these individuals, and then did a pretty intensive evaluation of their reservoir status, showing no recoverable virus from 150 million purified CD4 cells or from a rectal biopsy tissue sample from one of the participants. And so given the lack of detectable HIV despite pretty extensive sampling, both of these two Boston patients underwent a closely monitored treatment interruption, and then patient A at the top here was fully suppressed until day 84, excuse me, when they ended up having a detectable viral load of about 900 copies, and then a repeat test 4 days later went up to 127,000 copies. The patient was suboptimally adherent to ART, but eventually was able to be virologically suppressed again. Patient B, though, was able to be off ART for 8 months, which is actually fairly remarkable. If you recall that the majority of HIV-infected individuals have viral rebound within about 3 weeks, 3 to 4 weeks or so, and then subsequently had a viral load rebound, actually a pretty high one, up to about 1.9 million copies. So while these two participants did not reach kind of complete HIV remission, I think that they are incredibly valuable in teaching us some lessons. I think first, it shows that by depleting the HIV reservoir, this is by at least 3 to 4 logs here, you can significantly delay the timing of viral rebound. But without either HIV-resistant CD4 cells, and of course, sorry, I forgot to mention that both of these individuals received donor cells that were wild type for CCR5, right? So their CD4 cells from the donor was susceptible to HIV infection. So without either HIV-resistant CD4 cells or an active HIV-specific immune response, it does not appear to be stem cell transplantation does not appear to be sufficient to achieve long-term sustained cure. In addition, because there is about 100% donor chimerism, it means that the immune system was functionally naive to HIV. And what you're seeing here in the rapid viral rebound is what happens in acute HIV, including retroviral rebound syndrome as well. So, having mentioned the potential importance of the immune system, I did also want to mention that there are some non-reservoir predictors of HIV rebound that was found in 5345. Now, we are still kind of finalizing the analysis here, but I just want to show you one slide with some correlations of the non-reservoir measures. And in early treated individuals, you can see that there are some both T cell and soluble markers that were associated with either faster or slower viral rebound. For faster rebound, activated CD8 cells, and then a soluble CD163. And for faster viral rebound, you've got more higher percentage of effector memory CD8 cells, as well as CD8 cells expressing HIV-specific CD8 cells expressing CD107A here as well as some soluble markers. And so overall, just in summary here, a suite of HIV reservoir assays are being used in recent and current HIV cure studies. In 5345, smaller size and lower activity of HIV reservoir did predict a modest delay in rebound, but the strength of association really depends on the timing of ART initiation. The intact proviral DNA was strongest predictor for chronic treated individuals, and the ISCA was the strongest predictor in early treated individuals. Extreme reservoir depletion can lead to delayed HIV rebound, but residual infection will eventually fuel rebound without a HIV-specific immune response. And non-reservoir markers of HIV rebound and post-treatment control really should be explored. And I just want to thank my my co-chair, Davey Smith, as well as all of the 5345 participants, team, and site staff. So thanks.
Okay, thank you, John. And tantalizing biomarker data at the end there with the soluble markers, so fascinating, which is a great segue to our next speaker, Dr. Mohammed Abdelmozen from the Wistar Institute. we'll be setting our stage in terms of moving forward with these non-invasive assays and also non-viral markers for HIV reservoir measurements. Oh, turn it over, Mohammed.
All right, thanks, Tim. Can you see my slide in full screen and hear me well? Anyone?
We can see, yes.
Awesome, great, thank you very much. And thanks, Tim. And as you mentioned, the last point from John is a great segue for my presentation, which will focus on those non-reservoir measurements of a viral rebound. And I would like to start my presentation with this very simple question of why we do even need to identify non-invasive surrogates of the total body burden of rebound competent HIV reservoir. And as all of you know, upon treatment interruption, the vast majority of people living with HIV would experience a quick yet variable rebound. And the current intervention that aims to achieve ART-free remission and prevent this rebound would greatly benefit from ways to better understand this rebound and predict it. But this rebound is likely influenced by two things: the total body burden of inducible replication competent reservoir, but most of this reservoir reside in tissues. Some animal studies suggest that more than 98% of this reservoir is in tissues, and it's not clear to what degree studying the reservoir in the blood telling us about the reservoir in tissues, which is again about 98% of the reservoir. The second thing is the immunological pressure on this reservoir, again, in tissues where most of it reside. Now, we do not have access to most tissues, and even the one we do have access to, like the gut, maybe lymph nodes, there are major sampling and procedural biases that prevent us from accurately measure this reservoir or immunological pressure on this reservoir in these biopsies. So we need to try at least to identify surrogates for this reservoir in tissue and immunological pressure on this reservoir in tissues. So let's imagine this tissue with a small non-inducible reservoir. Perhaps this tissue will have a less inflammatory milieu compared to another tissue with a large inducible reservoir. Again, using biopsies from these two tissues, it's important to do quantitative, maybe even qualitative characterization of this reservoir, maybe reservoir-bearing cells, immune cells, using assay that is mentioned by Nicola and John and others. And this will be very important. But again, as I mentioned, this will be prone to significant sampling biases and other limitation. Perhaps something like whole body imaging can help overcome several of these problems, and depends on how sensitive this will be. And I believe we will hear more about that later today from Tim and others. But these two tissues with potentially different environment do release several biological molecules that some of them eventually will find their way to circulation by active secretion or leakage. And the quantitative and maybe the qualitative feature of some of those biological molecules maybe can tell us something about the tissues they are coming from and their inflammatory status. Now, these molecules could be cells, could be virus. Many believe that is residual viremia or reactivated virus coming from multiple tissues into the circulation. Could be antibodies. Likely the quantitative features of HIV-specific antibody is important, but even more important, likely the qualitative feature and their neutralization ability for autologous virus. This could be proteins, including inflammatory cytokines and chemokines, could be markers of tissue integrity. These tissues with different inflammatory status and immunological pressure will have a different metabolic burden, which can be also maybe reflected in different host and microbiome-related metabolite in the circulation. Could be lipids, glycans, exosomes, cell-free nucleic acid. Several other molecules also could be potentially measured in the plasma that can be coming from the tissues. And together, all of those molecules has been shown before in other diseases to be in a good marker of tissue environment, like in many tissue-centric diseases like gut diseases, heart diseases, neurological impairments, and they have the potential to be also telling us something about the status of tissues during HIV infection. That's in one hand, but in the other hand, some of those molecules are biologically active and they have the ability in their own merit to bind to a receptor on the surface of cells and starting a signal transduction that can impact cellular function. So while most of those molecules could be a passive biomarker, some of them could be also an active biomarker. And I will give you an example of some of those molecules. In our lab, we have been investigating whether host metabolite and glycans can be measured before stopping antiretroviral therapy, and their level correlate with time and probability of viral rebound after stopping therapy. And taking a long story short, as we have reported this in a couple of publications in the last couple of years, we analyzed sample from different cohort of analytical treatment interruption, and we identified some metabolite and glycan that is their level before stopping antiretroviral therapy associated with either a longer time to viral rebound or a shorter time to viral rebound after stopping antiretroviral therapy. And some of those metabolite and glycans can also differentiate who will achieve ART-free remission, or also called post-treatment controller, from non-controller. And when we combine some of those metabolite and glycans together in a multivariable logistic model, this model could differentiate post-treatment controller from non-controller using different statistical method with potentially high accuracy. So I want to quickly highlight a couple of things about these metabolite and glycan that can be applied for other potential non-reservoir measurement of rebound. The first thing is they were not randomly distributed, and they belong to a particular host pathway. For an example, the metabolite that their level before stopping therapy associated with delayed viral rebound were all embedded into anti-inflammatory metabolic pathway, including glutamate metabolism. While in the other hand, the metabolite that is their level before stopping therapy associated with accelerated viral rebound were all embedded into pro-inflammatory metabolic pathway, specifically those are that associated with the gut microbiome dysbiosis or translocation, including a high level of tryptophan catabolism. This is actually interesting because we have been seeing this over and over in multiple ATI studies, including the A5345 that is John described, that is high level of tryptophan catabolism is associated with accelerated rebound after stopping therapy, which is interesting because altered tryptophan catabolism has been known to mechanistically and directly impacting T cell functions and fate, and has been also associated with the development of several inflammation-associated comorbidities during HIV infection. And I think this is important because this is suggesting there are host pathways that are shared between the development of inflammation-associated comorbidities during HIV and HIV persistence or rebound. But it also suggests that some of those non-reservoir measurements could be reflecting immunological or inflammatory pressures that is associated with viral rebound and maybe directly or indirectly impacting it. And indeed, when we tested whether some of those metabolites can directly impact HIV reactivation, we found that as many of them don't, but some do. For an example here, I'm just giving you this example with this one metabolite that its level associated with delayed rebound and was higher in post-treatment controller compared to non-controller. And when we examined if this metabolite can directly inhibit HIV reactivation using both cell line model of HIV latency as well as primary CD4 T cells isolated from HIV-infected or suppressed individual, we indeed found that is this metabolite that associated with delayed rebound can inhibit the ability of BMA and amycin, anti-CD3, anti-CD28, and even some latency reversing agent to reactivate latent infection, consistent with what we found in vivo. So this suggests that this analysis could be useful in identifying host factor that might be important in posing some pressure during viral rebound, but which is in its own merit very important. But going back to the main goal in using those as marker for the total body burden of reservoir or rebound, there are several challenges and obstacles that will need to be addressed before we can entertain using this data for this goal. And I will highlight a couple of these challenges in my last slide here. Is we rely on analytical treatment interruption study to determine this potential marker. And by definition, these studies have very small number of participants and most of those participants experience a very limited window of time to viral rebound. Both of these two things are not ideal for this kind of analysis. Maybe you can combine different analytical treatment interruption studies together in order to increase the N and maybe increase the window of time to viral rebound. But analytical treatment interruption study has been very inconsistent in how to define viral rebound. Viral load threshold, how to define time to viral rebound makes it harder to combine different cohorts together. There is also a lot of inconsistencies in how to do these measurements. And something as simple as the anticoagulant can make a big impact on this measurement. So that's something also need to be addressed. Also, very little attention has been usually paid in analytical treatment interruption to account for clinical or demographic confounder. Clinical could be time to ART initiation, ART duration, ART regimen, pre-ART viral load or native CD4 count. Demographic could be ethnicity, age, sex, gender, and whatnot, which is all can impact those measurements. Longitudinal sample can overcome some of those, but usually they are not collected in analytical treatment interruption. And again, all of those challenges and other will need to be addressed before we can use these as marker. And I will just end with this point, that is, when we address these challenges, it still will be unlikely that is a single or even a handful marker can predict these complicated virological and immunological measurements. And we will likely need a combination of both viral and host marker to be trained together to generate this signature. And I will just end with my acknowledgement slide, and thank you.
Excellent. Thank you, Mohammed. Fantastic. And we will move right along here to Dr. Matthias Lichterfeld, also from Harvard, speaking us today. And Matthias will talk to us about single-cell analysis technologies and assays that he has been shown recently to be extremely helpful and informative on measuring HIV persistence and residual burden. So, yes.
So, hi everybody, it's Matthias here. Can you hear me?
We hear you and see your slides, yes.
Perfect, yeah. Good. So, yeah, I'm going to talk about 4 different assays today that all are designed to look at single HIV reservoir cells. The sort of a key feature of these assays is they all can look at these reservoir cells directly ex vivo, and they are sort of designed to provide complementary pieces of information about these reservoirs. So the first assay is the FlipSeq assay that Nicolas has already talked about. I'm not going to say a lot about it, but it simply works by doing limiting dilution assays, near full-length amplification of proviruses, and then next generation sequencing, and then doing a bioinformatics analysis at the end of this. And what I just want to emphasize is that these assays can certainly allow us to distinguish intact and defective proviruses, which is the key parameter, but we also get so much more information from these studies. We can look at clonality, footprints of CTL immune selection, footprints of BNAP resistance mutations, tropism, drug resistance mutations. So really a lot of information that we can get out of these sequencing data. And in the past, these types of information has been collected sort of from random DNA, and now we can do it by looking at the intact proviruses, which makes this a lot more informative. The next, and we can also use this assay for looking into very selected patients and figure out if they have any intact proviruses at all. I'm just showing 2 examples for this here from our work, where we have looked at billions of cells and never found a single intact provirus. So that then brings up the possibility, is it really a sterilizing cure, a virological cure that we are dealing with in these patients? The next assay that I wanted to talk about is what we are calling the MIP-seq assay. This is an assay that's designed to analyze the proviral sequence and the corresponding chromosomal integration sites from single proviruses. The workflow is shown here. So we are doing limiting dilutions again, and then we are doing whole genome amplification, and then we are doing dedicated assays for looking at the near full-length proviral sequence and the corresponding integration site. The reason for doing this is really that human genome that we have is incredibly complex. And from our perspective, it makes really a lot of a difference if HIV is sitting in certain parts of the genome that are transcriptionally active, or in parts of the genome that are not active. And it's actually interesting to recognize that only 20% of that genome that we have is genes, so is encoding for proteins. 80% of the genome is not really involved in gene transcription. And if HIV is sitting in those regions, that probably makes a lot of a difference. So by using this assay, we can distinguish proviruses that are located in genes, which is the ones that are not located in genes. We can also look at repetitive genomic elements, and we can also look at the configuration of them. Proviruses. And what makes this assay particularly informative is if we take advantage of all this data on the human genome that already exists, specifically the epigenetic data, and we can use that to calculate distances between proviruses, and specific epigenetic features. What we are also proposing is that these integration sites can be used as biomarkers of selection processes that are occurring in the reservoir. And one example for this is shown here for an elite controller, where this has been picked up most definitively. So you can see here in this phylogenetic tree here, each of these symbols reflects one intact provirus, and the reservoir in this person here seems to be really just composed of two large clones. And one of them is integrated into that centromeric satellite DNA here on chromosome 7. And here the second clone is in a ZNF gene on chromosome 19. Both of these are heterochromatin region, and it raises this sort of provocative hypothesis that these proviruses, these clones can persist long-term because they are sitting in parts of the genome where they cannot be actively expressed, and then the immune system is not really targeting them. But at the same time, all these other proviruses that are sitting in more accessible parts of the genome, those presumably have been effectively eliminated. That's our hypothesis. We have recently extended this work and looked into persons who have been on ART for very long periods of time. This is an example for one of these studies. So this is a person here who was sort of identified and diagnosed with HIV back in 1999, and then started treatment around that time, and has been continuously on ART for the subsequent 20 years. And we are looking here at the each of these symbols here is reflecting one intact provirus. And we can see here at an early time point back in 2000, most of the intact proviruses that we pick up are located in genic locations. There's one big clone here of intact proviruses in that ITGAL gene. Then after about 10 years later, we can see that this ITGAL clone here is still persisting. There's one more clone here, but then there's also the centromere clone showing up here. And then after another 10 years, that centromere clone is actually dominating the entire reservoir profile, and all these proviruses in genic locations are no longer detectable and presumably have been eliminated by immune-mediated mechanisms. So this is sort of the idea that we are proposing integration sites as a way to track selection processes that are occurring in the reservoir, and that we can identify. Just focusing here on the same patient, looking at the defective proviruses from that very same patient. And you can see here for the defective proviruses, it looks quite different. We see defective proviruses all over the place, a lot of work that has been invested here. But we don't see as much of a selection process that we saw for the intact proviruses. And it raises this possibility that the defective proviruses are just behaving very differently and are not as much under selection pressure as the intact proviruses. Just going to talk about one more assay, which is called what we call PRIP-seq assay. So this is an assay to get to this point of HIV latency or HIV transcriptional activity. So again, we are doing this by diluting reservoir cells to single genomes and then or single HIV infected cells. Then we are extracting the genomic DNA and the RNA. The genomic DNA is then sort of put into this MIP-seq workflow, so whole genome amplification, proviral sequencing, and integration site analysis. And the RNA is used for reverse transcription, and then there's cDNA amplification, and then we can quantify individual HIV RNA transcripts on a ddPCR instruments. And at the end of this experimental workflow, we have these three different components, so the proviral sequence, the integration site, and the transcriptional activity from single HIV infected cells. Just an example of how this looks like, we did this in a large study here where we included about 701 individual reservoir cells from 6 ART-treated patients. And one of the important conclusions from the study was that chromosomal location is very much connected to transcriptional activity of the proviruses. So if a provirus is located in a gene, then it's in 30 to 40% of cases, these types of proviruses actually are producing viral RNA. However, if these proviruses are located in non-genic DNA, specifically in satellite DNA, then the probability that they are producing viral transcripts is very low. It's maybe in the range of 5%. So that's why we are coming up with this proposal that proviruses here in these non-genic satellite DNA regions are in this condition that we are calling deep latency. And at the same time, proviruses that are located in genic positions are not necessarily latent. They tend to be transcriptionally active in a significant proportion of cases and can presumably be detected by immune mechanisms. That's our hypothesis. So that's just the PRIP-seq assay. We believe it's very helpful to look at the transcriptional behavior of individual proviruses, and it sort of reflects the susceptibility of proviruses to in vivo, to physiological viral reactivation signals. We believe this is helpful for looking at the depth of latency. And it sort of challenges a little bit this concept that we are dealing with latently infected cells. And in many cases we do, but in other cases we don't. And there's a considerable proportion of proviruses that can actually be transcriptionally active despite long-term ART. Briefly going to talk about the last assay that we have recently developed. This is called PHEEP-seq. So this is an analysis of the proviral sequence in combination with the cell phenotype. And we believe this is helpful and informative to learn something about the immune susceptibility of HIV reservoir cells. So the workflow here is that we are starting with ex vivo isolated cells. We do a memory cell isolation by negative mechanisms, and then we label these cells with antibodies that are barcoded. And then these cells are entered into this machine that does single cell encapsulation and cell lysis. And then the cells are being barcoded. The DNA is barcoded so that DNA from each cell can be identified. And then we are using multiplex single cell PCR to amplify selected parts of the HIV DNA and of the antibody tags that were used for cell labeling. At the end of this process, all these amplification products are pooled, and then they are put on a next generation sequencer. And then there's bioinformatics work done to identify the sequencing reads that originate from individual cells. This is just the way how we have designed this. So this platform supports amplification of small fragments, about 200 to 300 base pair fragments, but you can do a lot of those here at the same time from a single cell. So we ended up designing primer pairs that amplify 18 different fragments that are positioned over the entire HIV genome. We have the IPDA primers integrated into this assay here. And at the same time, we also have primers in place that are designed to amplify viral host junctions of large clones that we have previously identified in our patients. We can combine this with a total of 53 different antibodies that are being used for surface phenotyping, allowing us to do a relatively comprehensive phenotypic analysis. This is just an example of how this looks like here from one particular patient. So we sort of separate the cells in different categories. We have this category 0, which are essentially all HIV infected, all sort of HIV non-infected cells here. And then we have what we are calling category 1, which are all the HIV infected cells that have any type of HIV DNA. And we also have here this category 2, those are cells that harbor intact proviruses. And then we have category 3, those are cells that harbor clonal proviruses. And what you can see here is that there are quite a bit of differences when we compare specifically the cells that have intact proviruses. So this here, category 2 towards category 0. And that's particularly true if we look at some of the markers that are related to immune checkpoints or exhaustion. So this is particularly BTLA, KLRG1, PD1, and TIGIT. They tend to have relatively limited expression in HIV non-infected cells, but in these cells that harbor intact proviruses, it's actually higher. Also, these markers here, HLA-E, HVM, PD-L1, and PBR, those are markers that are sort of delivering negative immune regulation signals to effector cells, sort of delivering a message of do not kill me. Those also tend to be higher in cells that harbor intact proviruses here relative to the ones that do not have any HIV DNA in it. We also looked at certain cytokine receptors, particular CD127 seems to be different between those two cell types. IL-21 receptor seems to be different, and also the TGF-beta receptor here seems to be different. And also CD49D, which is an integrin and associated with immune activation, also seems to be different. So a very high resolution analysis of the phenotype that's possible with this type of technology. What's also built in is that we can look at individual clones. So here we have a list of individual clones that we looked at. All these clones are identified based on chromosomal integration sites. Some of them are intact, some of them are defective. And we can look at the individual phenotype of these clonal cells. Overall, I'm not going to go into the details here, but we see a relatively unifying pattern that many of these immune checkpoint markers and these negative immunoregulation markers tend to be upregulated in these HIV-infected cell clones relative to HIV-uninfected cells shown here. So that's just the summary on the FEEP-seq assay. I believe it's one of these assays now that is available for looking precisely at the phenotype of infected cells. We are doing this directly ex vivo, so there's no tissue culture artifacts. We can do this and distinguish cells that harbor intact versus defective proviruses. We can do this in cells from blood and from tissues. We have done this here with 50 phenotypic markers, but now we are in a position that we can greatly expand this and do a much more comprehensive analysis. And what this assay is telling us is a little bit about what's the susceptibility and the vulnerability of these reservoir cells. What type of resistance do they have potentially to certain immune mechanisms? And we believe that's particularly informative when we discuss immune interventions for reducing the viral reservoir size. Overall, I believe there's certainly great progress in developing assays for reservoir cells in the last couple of years. As you can hear in this meeting here, it's great to have the choice of assays for specific biological questions. And I believe we can take advantage of these assays also for looking into clinical trials and really make those studies most informative. Just going to sum up here by summarizing our collaborators who are really doing an incredible job in contributing, and our team here in Boston from the Reagan Institute and the Brigham and Women's Hospital. So thanks again for inviting me. And that's it from my side.
Excellent. Thank you, Matthias. Fascinating. I love this last assay. This is going to be very important going forward, I think, for sure. So thank you for presenting that data. And last in our session, is going to be Dr. Sarah Palmer, also needs no introduction. And I apologize for running a few minutes late. And I believe Sarah will have a pre-recorded talk, given that it's almost 1 a.m. or so, I believe, in Sydney right now. I apologize. And then we will follow up with a Q&A session directly after the talk. And I believe that Dr. Palmer will be on to answer questions heroically. So I will now turn it over to the pre-recorded talk.
During effective antiretroviral therapy, HIV persists in several anatomic sites and different cell subsets. In order to develop effective curative strategies for this persistent virus, we must answer the question, where does HIV persist during therapy? What cell types and cellular mechanisms contribute to persistent HIV during effective therapy? And which cells contain genetically intact replication-competent HIV? To address these questions, full-length HIV DNA sequencing methods have been developed by laboratories across the globe. And the application of these full-length HIV DNA sequencing methods has given us some clues as to where HIV persists during therapy. We applied our full-length HIV DNA sequencing assay to untreated participants who were enrolled in a clinical study in Bangkok, Thailand. And this was done in collaboration with the Thai Red Cross AIDS Research Center. And we sequenced the provirus and the PBMCs from these untreated participants. And there was 2 cohorts of participants which we sequenced their virus. It was a HIV co-infected cohort and an HIV mono-infected cohort. And what we found is that there were high levels and a high number of genetically intact proviruses in the PBMCs of these participants. In fact, 60 to 3,000 genetically intact proviruses were identified in 1 million CD4 T cells. And to put this into context with what we see in patients who are on antiretroviral therapy, is that typically we will sequence 0 to 26 genetically intact proviruses per 1 million cells in patients who are on effective therapy. This indicates that when patients are during pre-therapy, that their PBMCs can contain high numbers of genetically intact proviruses. In contrast, when Yachi Ho and Katie Bruner applied their full-length sequencing assay to PBMCs from patients who were on effective antiretroviral therapy, they found that the majority of proviruses in these PBMCs were genetically defective. In fact, they found that these PBMCs contained only 2% of genetically intact proviruses. Bonnie Heiner and Guinevere Lee analyzed full-length individual proviral sequences in cell subsets sorted from patients on effective antiretroviral therapy. And what they found was that these cell subsets also contained genetically defective proviruses. In fact, only 5% of these cells, or 5% of the proviruses found in these cell subsets, were genetically intact. However, our research team and other research teams have found that genetically intact proviruses are unequally distributed in CD4 T cell subsets. In fact, we have found that effector memory cells and effector memory cells that express the activation marker HLA-DR contain significantly higher levels of genetically intact proviruses compared to other cell subsets. In a recent study by George Waymer et al., they sorted quiescent latent memory CD4 T cells based on their specific antigen receptor, and they mapped the transcriptional profile of these cells. And what they found was that proliferating CD4 T cells, which contain intact proviruses, have a transcriptional profile that is predominantly effector memory. In fact, they found this for the majority of their patients, that those cells that are proliferating and contributing to genetically intact proviruses are effector memory cells. So we wish to look to the proviral landscape to determine if this proviral landscape will give us some clues as to why we find genetically intact proviruses concentrated in effector memory cells. Gabby Duette and Bonnie Heiner, our group, analyzed these proviral sequences from 24 ART-suppressed participants who were on ART for 2 to 22 years. They compared the genetic landscape of persistent HIV between these cell subsets that were sorted from these participants. And what they found in agreement with our earlier studies, that genetically intact proviruses are concentrated in effector memory cells. They also looked at defective proviral landscape across the different cell subsets. And what they found was that 5' deleted proviruses, and these are proviruses with a large deletion in the 5' end, but are genetically intact in 3' end, are concentrated in effector memory T cells and significantly concentrated in effector memory T cells. They found that 3' deleted proviruses, which contain a deletion in the 3' end, are significantly reduced in effector memory cells, and that those proviruses that have a large internal deletion were also significantly reduced in effector memory cells. So this indicated to us that some genomic part or region of the provirus at the 3' end allows for HIV proviruses to persist in effector memory T cells. So we sequenced a lot of the open reading frames across the genome, and what we found was sequences with intact NEF open reading frame were concentrated in effector memory cells and significantly concentrated in effector memory cells compared to other cell subsets. This is very interesting to us because the expression of the NEF protein downregulates MHC, making HIV-infected cells essentially invisible to CD8 T cells and resistant to CD8 T cell clearance, and allowing these HIV-infected cells to persist. When we looked at sequences with intact GAG and NEF open reading frame, we found a high percentage of these sequences with an intact GAG and NEF open reading frame were significantly, again, concentrated in effector memory cells. And this indicated to us that an intact NEF open reading frame could protect other genomic regions of the provirus, particularly the GAG region, which is highly immunogenic. Another question we need to address when developing effective curative strategies is, how do plasmid variants contribute to persistent HIV? How does pre-therapy plasmid-derived variants contribute to the HIV reservoir in cells? And what does rebound virus look like, and which cells contribute to this rebound virus? To address this question, Katie Fisher and our group developed plasma-derived HIV RNA long-range sequencing, or the PEARLS assay, allowing Katie to sequence an 8.3 kb or 4.7 kb region of HIV RNA. When Katie applied the PEARLS 8.3 kb assay to 8 participants during untreated infection, she found that 65% of the plasma-derived genomes were genetically intact. So this indicates that up to 50% of viral particles in the plasma contain defective HIV RNA. And the most common type of defect in this RNA are frameships, and that's followed by deletions of a size greater than 100 base pairs. Katie also applied her PROLS assay, sequencing the 4.7 kb region, to three patients who underwent an analytical treatment interruption. And she compared the sequences from the analytical interruption plasma to pre-therapy plasma sequences. And what she found was that the proportion of 100% identical sequences was higher in the ATI plasma sequences at 30% compared to the pre-AR plasma sequences at 13%, but this did not reach statistical significance. Katie also found, for these three participants undergoing an ATI, the phylogenetic analyses revealed that ATI plasma-derived sequences and ATI plasma-derived sequence was 100% identical to a cluster of pre-ART plasma-derived sequences and PBMC HIV DNA-derived sequences from pre-therapy PBMC samples. This indicates to us that the reservoir developed during pre-therapy can contribute to rebound virus during an analytical treatment interruption. So in conclusion, our sequencing analyses have revealed that there is a difference in the proviral landscape between cell subsets of memory CD4 T cells within the peripheral blood. Genetically intact proviruses appear to be concentrated in specific memory T cell subsets. Cellular proliferation contributes to HIV persistence during therapy. Cells which are more proliferative contain more genetically intact HIV. Not all variants in the plasma are infectious. In fact, up to 45% are defective. The majority of ATI plasma-derived sequences are genetically intact. Several sequences from the ATI plasma samples were identical to viral sequences from pre-ART plasma and PBMC samples, indicating that HIV reservoirs established prior to therapy contributed viral rebound during an ATI. In spite of the breakthroughs in knowledge we have achieved based on these technologies, our ability to use this technology around the world is hampered by a number of factors. First, these sequencing techniques are expensive. Second, technological know-how may be lacking in different parts of the world, requiring significant investment in education, training, and capacity building. Third, concerns within industry as to intellectual property rights protections could slow the rollout of these technologies, especially in countries where such protections are weak. So I would like to thank the large group of people in my lab who contributed some of the data to these studies and presentation. I'd like to thank our collaborators, the Kirby Institute and the Department of Medicine at UCSF, Steve Deeks and Becky Ho. I'd like to thank our funders, but most importantly, I would like to acknowledge with gratitude the participants of our studies. Without their participation, we would not be able to conduct our research. Thank you.
Excellent. Thank you, Sarah. And we now have time for about 10 minutes until it'll be 7:20 a.m. my time, which we'll have a 10-minute break, after which Bonnie will take over to lead some of our flash talks, followed by our breakout room discussions, which will be very important. But I wanted to, we have a few minutes now. If folks have any directed questions to either or all of the speakers, feel free to either raise your hand or put your messages in chat, and we can kind of facilitate some of the discussion. I don't see anything in the chat right now, so maybe I will. Oh, wait, Nicola has a question, so why don't we hand that over to Nicola to start.
Yeah, thank you, Tim. I have a question for Sarah. And Sarah, I'm sorry because I know you're in the middle of the night right now, but so feel free to actually type your answer if you prefer. So I was wondering whether you tried to use your beautiful sequencing assay of plasma viruses in people on suppressive therapy. So basically, do you have the ability of sequencing these few viral particles that are still circulating in the blood of people on fully suppressive ART? And if yes, is it actually a better predictor of the virus you would see during rebound?
Yeah, that's a great question, Nicola. Of course, we're always limited by the amount of RNA in the plasma when patients are on effective therapy. We have applied the 4.7 kb sequencing region to plasma with patients on effective therapy, and we have been able to sequence that region. Sequencing the 8.3 kb region is a bit more challenging. But yes, I would agree that it would be really important to sequence the virus during therapy, and especially using long-range sequencing so that we can really identify what virus during therapy is contributing to rebound.
And Victor, it's your hand up as well.
Thank you. Yeah, sorry about that. I found your presentations fascinating, Sarah, and one thing I found very intriguing was the potential role of NEF in making the reservoir stick around. So you have these NEF sequences. Have you had a chance to confirm that those proteins actually have the function of down-modulating MHC class 1, the ones that were enriched and the ones that you saw?
Yes, so in our publication in JCI, the recent publication in March, we did actually test whether MHC is affected by HIV isolates that are NEF deleted versus those that contain NEF. And yes, absolutely, those that contain NEF down modulate MHC, and those that are NEF deleted have more MHC expression.
But was that in the context of a provirus or in the context of a NEF clone NEF gene?
That was in the context of a lab strain, that 2 lab strains, one that had NEF and one that was NEF deleted.
So my question was, do the sequences that you have that activity? Because this means in general, NEF does have that activity, but primary isolates of NEF vary on which activities they preserve. So I was wondering if those ones that you found specifically had that activity in the context of the protein itself, because in the context of the provirus, there might be intervention of other proteins that might alter cell surface receptor expression.
So to be honest, no, we have not done those studies.
Okay. Well, if you're interested in doing some of that, both in vitro and in vivo, let's talk after the call.
I probably won't be on the call, but we can talk via.
No, that's figuratively speaking.
Okay.
That's a long night. I saw Matthias and then Alan with their hands up.
Yeah, I just had a follow-up question for Sarah. You said that the plasma virus in people who are doing ATIs is more likely to be intact compared to people who are not doing ATI. Is that true? Yeah. Can you explain
we
that a
found
little
about
bit?
87. Yeah, well, we did the full-length sequencing, and we're doing that now for all. So this is a very interesting cohort of participants that we're sequencing right now. They've had 3 different ATIs. And we're sequencing all 3 ATI time points using our full length or 8.3 kb sequencing, can't really say totally full length. And we are seeing that about 87% of the viruses during the ATI are genetically intact versus pre-therapy where we saw there was a lot more defective. Yeah.
Is there any good explanation for that? Or?
Only that maybe those cells that are producing this virus must have replication competent virus. Yeah, that's the only thought I have. It's very interesting.
Yeah, Alan and John.
Great, thanks, Tim. Really wonderful talks. Let me approach something to everybody, but maybe focus on John Lee. I mean, we've heard a lot of the wonderful science and approaches we've taken from the viral and immune. And John, since you think a lot, you're involved a lot on the clinical trials side for the CTG as a laboratory virologist. How would you take what you've heard today so far and integrating it into the clinical trials realm? I think we do a lot of great pathogenesis studies, but at the end of the day, this is the industry academic collaboratory of where we want to move the field forward and applying these tools in our clinical trials and using them effectively in understanding the role of cure studies and outcomes. Do you have any, and everybody talked about the limitations and seeing all that through there. John, any larger thoughts or how we decide?
I mean, I think we're still in somewhat of a discovery phase when it comes to all these reservoir measures. Sometimes I do feel like, that old parable of all these the blind men feeling the elephant. One assay is looking at the tails, and another assay is looking at the trunk. And if we just focus on one assay, you're going to miss a potential overall picture. And so I do think that these trials that are looking at the whole suite of reservoir markers, I think at this point it still makes a lot of sense. And I think the newer assays that Matthias was describing is tremendously exciting. The single cell assays and also Sarah's ability to do full length proviral plasma sequencing, I think is something that I'm particularly interested in. We've been doing a little bit of that in our lab as well. We're able to kind of sequence about an 8 kb region as well, especially with our recent finding in 5345 that the single copy assay was associated with timing of viral rebound in early treated individuals. Really does speak to me that there might be something qualitatively different in the residual viremia of early treated versus chronically treated individuals. So I think this is an exciting time for studying the reservoir because the technology is finally catching up with the questions that we want to answer. Anyway, that's what I have. I'm sure others have other thoughts as well.
And how would you incorporate, as we've done in 5345, the immune or other suite of assays that Mohammed also spoke about and has done in some of the 5345? Do you see all these as complementary to each other and understanding the virus and the host at the same time?
For sure. I mean, it's Mohammed's data is also very exciting. I think that, as I said, the reservoir measures are not perfect, but you might be able to have a surrogate marker that is a reflection of key aspects of the reservoir that might be even better than measuring the reservoir itself. We just don't know yet. And also it's, downstream consequences of reservoir activity or reservoir size that, especially within the tissues, right? We can't measure the tissues very well, but maybe with some of these non-invasive markers that might not even be virus specific, it might be a better reflection of the whole body reservoir and the whole body activity reservoir that even our single copy assay or our IPDA might of the peripheral blood might not have as good of a view on what's happening in the tissues as with some of these other non-virologic, non-invasive measures that Mohammed's talking about.
All right, thank you. John, that was a great actual wrap up to the entire session actually as well. And I think those take home points are extremely important. And I think I just want to drive home because we have to break now for 10 minutes. Is that the technology is certainly improving rapidly, and we're now getting more and more tools. And so, regardless of what study is done, as long as we're carefully collecting samples, including tissues, if we can, for future studies, this is going to be key, I think, in those clinical studies as well, to make sure we have sufficient material and the types of materials we need to do these types of assays as they come forward. So, thanks everyone for this first session. Please come back in 10 minutes. Where Bonnie will lead off a few flash talks on non-invasive measurements of the viral reservoir throughout the whole body, followed by our breakout rooms, which will be very important in terms of having discussion, in fact, after which we'll have a callback. So 10 minutes and we'll be back. So please rejoin. I'm going to go caffeinate.
Well, welcome back, everyone. Tuli, are we about ready to get started, or do we need a few minutes?
I believe we are ready. So it's 4:30. I think we can go ahead and start with the next session.
Okay.
Yeah,
no, that's great. And just verifying that Tim is online because he is next to speak. Tim, are you there?
I am here.
Oh, awesome. Okay, terrific. Well, so now we're going to proceed into the short presentations of these flash talks. They will be 7 minutes each. Again, we encourage you to think about questions and put them in the chat. And we will also, at the end of this session, have a 15-minute block of time where we can ask questions of the speakers directly. And so we'll start off with Tim, who, as you know, is from the University of California, San Francisco. And we will follow up Tim's talk with a talk by James McMahon, who is an infectious disease physician and HIV researcher at the Alfred Hospital and Monash Medical Center in Melbourne. And so both Tim and James will be addressing the topic of whole body imaging and how they're using this new capability in their intervention studies. So I'll turn it over to you, Tim.
Great, thank you. And I just want to make sure people can see the reviewer mode here on the slide.
We can see it, but we see both views. So we just need you to put it in full screen present. Yeah. Change your display settings. Yep.
This one here. Okay, how's that?
Perfect.
Perfect, okay. Excellent, and thank you, everybody. This is the flash talk, so I will be fast and flash. And so I'll be talking first here about whole body tissue imaging of the HIV reservoir and immune responses. And as those of you likely know, we've been working on a lot of immuno-PET or positron emission tomography approaches to imaging the HIV reservoir using monoclonal antibodies directed towards the GP120 portion of HIV. And I think these PET imaging approaches can really have several applications and can answer several important questions. So for example, where does HIV persist throughout the body? Throughout the tissues during different stages of infection, for example, before, after ART. What are the dynamics of HIV decay on ART when someone starts? And when I say dynamics, I'm looking at the regional and temporal dynamics of decay. And what tissues and anatomical regions are responsible for HIV rebound following cessation of ART? Obviously, we can also ask what tissues and anatomical regions are responsible for reactivation in the setting of, for example, LRA therapy or blocks to HIV latency. And obviously, this will hopefully ultimately perform a platform to measure HIV activity and burden in various HIV cure clinical studies that can actually have a direct practical use with clinical trials. Just to briefly recap what we've published thus far using a VRC01, our first pilot, using a long-acting 3-day half-life zirconium radiotracer. And seeing that we could see an intake of tracer in inguinal lymph nodes, particularly those that were viremic, but also low-level yet subtle signals in lymphoid tissues and other tissues in ART-suppressed individuals. But really, we see very little uptake in those that are uninfected in kind of age-matched controls, lifestyle-controlled participants. And just to look a little bit more detailed to see that, again, we see that these uptake values, which are standardized across the size of the participant, the amount of dose, the timing of the dose that was given, et cetera, we see that there's significant increases in inguinal lymph nodes, for example, in some of these uptake values compared to uninfected controls, but also in those on ART. We also saw this in the setting in sigmoid colon and in other tissues as well. So I think this really provided a platform where we were able to visualize minimal residual activity of HIV reservoirs in the setting across the body, with exception with the CNS and central nervous system. We've also been now using a high sensitivity massive parallel PET machines called Explorer. This is a super PET that allows us to image with high amounts of resolution, but also increase our signal to noise and the sensitivity of detecting tracer throughout the body, which is currently ongoing. And we have initiated this here. This machine actually is now FDA approved for clinical diagnoses. And I think over the next 10 years or so, will really become the forefront of clinical PET imaging, not just for research as well. So hopefully we'll see how these increasing technologies can really help with these types of immuno-PET imaging techniques. Well, we've also been looking not just in folks across time and cross-sectionally at those on ART or people that are viremic at the time of sampling, and found that we wanted to look at rebound dynamics using PET imaging. This is an example of someone that was in Lily Cohen's GATE study of a 64-year-old cisgender woman with HIV on ART, was actually imaged on ATI day 19, so 19 days after they had ceased taking ART. And this is 3 days after we injected the tracer. So we inject the tracer, wait 3 days, and then we do our first of several series of images using both PET MRI, which I'm showing here on the left, as well as Explorer imaging. Plasma viral load was not detected at the time of injection and at the time of this image that was taken. And then the following day, we saw a rapid increase in rebound of viral load. And what we're actually seeing is uptake not just of the liver, which is just the metabolic process of the monoclonal and the tracer itself, so that's just background noise. But we saw a marked uptake in bone marrow, spleen, various lymph nodes, the NALT, as well as the colon and other parts of the intestine. And we actually saw this in patchy areas. So it wasn't a kind of a general distribution like we saw in some of our people with HIV on chronic ART, but really patchy areas across different lymph node chains and within the bone marrow as well. So we're seeing a little bit different uptake here as we go through. So this is an example of how we can use this technique to look at dynamics of reservoirs during various interruption studies or other clinical trials. And we say that we actually do serial imaging every several days as well, so we can get a temporal dynamic of this rebound using non-invasive imaging. Well, you may say, why is this important? How can we use this practically in cure trials? And that's why we're here today to talk about this. And I think that this recent publication from Sadie Sammer with Tom Hope and Francois Villinger and Elena Martinelli, using immuno-PET imaging, using a similar technique to look at differences in HIV activity in macaques before and after blockade of TGF-beta signaling, which actually reactivated HIV-1 reservoirs. And you can see here on the left versus the middle versus the right that there was an increase in gut uptake of tracer as well as lymph node uptake. I think you can see one in the axillary lymph node there as well, following TGF-beta blockade. So very interesting that you can actually see throughout the entire tissues an impact of this. And these are things that you may miss, for example, in the peripheral blood. And just to give you an idea, again, in images from this paper, which is not, oh, here we go. Over here, you can also see that before and after TGF-beta blockade, a dramatic increase in gut wall signal as well as in lymph node tissues as well. So just an example of how this could be used going forward. Well, it's not just HIV specific, but I think following up on what Mohammed was talking about earlier, that you could also look at non-viral markers or other indirect measures of looking at viral persistence. And one of those is we're actually looking at we're trafficking T cells that are activated in trafficking using a guanosine analog that's fluorescently labeled to a fluorine 18, which is a very short half-life, but we're able to get very high resolution of images of where activated and migrating T cells are within tissues. And we've seen that in various individuals with and without HIV and with and without spontaneous control of infection, that we see differential uptake of T cells in various tissues, not just in lymphoid tissues here, but also in oropharyngeal tissues, glandular tissues, bone marrow as well, like we were seeing with our VRCO1 imaging as well. And so this is yet another technique that we can use, not just to look at the direct reservoir, but looking at tracking of immune responses within those tissues. And lastly, folks here at UCSF, and we're working in collaboration, this is now being implemented in those with cancer, but we cannot just look at where T cells are trafficking, but also if they are producing granzyme B. So can we actually image the functional response or T cell or antiviral response in the setting of various interventions? And this could also include vaccine trials or immunotherapeutic trials that may increase CD8 T cell function for trafficking to tissues to eliminate viral reservoirs. And this essentially uses the granzyme itself to cleave a very inert molecule, a helix formation that then binds to the cell membrane, has no biological activity, but just basically gets stuck on cells adjacent to where the granzyme B is being produced, and then we can image those with PET imaging. So just some examples of what we could do. Other groups have also looked at CD8 T cells and CD4 T cell imaging in the setting of SIV, and that have also been used to show dynamics of immune responses during various HIV curative studies. And so I just wanted to finish up really just to talk about some upcoming applications and to kind of foster some of the discussion during our breakout. This could certainly be used to look at elite exceptional control, not just looking at HIV persistence or residual GP120 production, but also looking at immune responses and how CD8 T cells penetrate tissues and if they have functional capacity within those tissues as well. Can also determine early sites of viral responses during ATI. And obviously these can be applied, I think has been shown with the SIV model so far, to a variety of cure-related clinical studies and ATI protocols. And I think that, going back again to what we're saying, that tissue-level studies are also critical and crucial in order to understand what we're seeing with these non-invasive methods. For example, are we seeing free virus that's being released by not just transcriptionally active cells, but virion producing cells within local tissue? Are we seeing GP120 being expressed as antigen on the surface of cells, probably at low levels would be my guess? Or are we seeing immune complex deposition or local production of these complexes within the tissues as well? And I think these are things that we still have to fully understand going forward. But these are certainly some of the challenges and some of the promises of these non-invasive imaging techniques as we go forward. And with that, I will hand it back to Bonnie.
All right. Thank you. Terrific. Thanks, Tim. And anytime you say that you're going to give a flash talk and talk really fast, just scares me, but that was perfect. Did an excellent job. Okay, so now we will have a pre-recorded talk by James McMahon.
Many thanks for the introduction. My name is James McMahon, an infectious disease physician researcher in Melbourne, Australia at Monash Uni and Alfred Hospital. Very honoured to be invited to speak today about the clinical trial we conducted on imaging HIV in vivo, and I'll start my slides. So essentially, this audience will be well versed with the need for methods to safely image and quantify HIV in tissue sites, and this is what we endeavoured to do with this clinical study. Of note, there was the work by Santangelo et al. Showing this was performed in a macaque model of HIV infection with a SOV NAb bound to copper-64 isotope. We also obtained 3BNC117 antibody and chelate with Mikasar, and using the same isotope, performed some experiments to understand that this still maintained its activity in vitro, and then in the clinical trial was of HIV uninfected and infected individuals from infusion of 3 mg/kg of the antibody with copper and imaged over 2 days. The assays was an ELISA initially to understand that the Mikasar bound antibody was no different to unlabeled, a cell binding flow cytometry assay with different envelope proteins expressed on cell surface, again showing no different on flow, whether it was unlabeled or unbound antibody or combined with Mikasar, and a neutralization assay, again looking at multiple different strains of envelope on a pseudovirus assay, showing that this neutralized these pseudovirions, whether it was just the antibody on its own or the antibody combined with Mikasar. The summary, we shows a 3 mg/kg dosing based on linear PK in the work by Kasky et al. And then used that combined with a smaller amount of the antibody bound to copper. So this is a combination of cold unlabeled antibody and hot antibody, very commonly performed in oncology. And this model also enabled us to, using copper-64, image healthy uninfected individuals with a total dose of less than 5 millisievert, which was required at the time, but also consistent with the macaque model. And work that we would be able to see imaging over that uptake over that first 48-hour period. We enrolled adults, either uninfected or HIV infected, on or off antiretroviral therapy, and the viremic group had a viral load above 1,000 copies, and the suppressed individuals were on treatment. We used 2 assays to understand if the participant virus maintained sensitivity to 3BNC, an outgrowth assay, neutralization performed at the Lewin lab in Melbourne, and at the Monogram bioscience assay performed by using a neutralization assay on participant-derived envelope. Whole body imaging was performed at PET-MR, and the first 2 individuals were recruited, and infected individuals were enrolled, and then the first 2 people that were viremic. Those viremic individuals, the uninfected, this is the PK of the uninfected individuals, showing no difference between an ELISA to assess the concentration of the antibody or radioactivity. But when comparing to the first 2 viremic participants, we could not see difference between uninfected and infected individuals, but we're confident that the chelator and isotope were still well connected to 3BNC. We amended the protocol to only use a small fraction of antibody, or just hot labeled antibody only injected, so less than 5 mg, and the radiation was increased based on dosimetry of the first participants enrolled. Ultimately, 4 viremic individuals were imaged with tracing or just hot antibody, some aviremic individuals, and also we repeated this with the uninfected controls. We had good concordance in the screening assay between the viral outgrowth and the monogram assay for identifying individuals with sensitive virus. And there's some of the images, the PET image, whole body PET images. This is the hot and cold dosing. This is the hot dosing only. There was increased uptake in the spine in only the small fraction of antibody that was hot labeled. And we progressed forward to further viremic individuals. But ultimately, we did not identify differences in the uptake between uninfected individuals. This is PET and some fused PET-MR images, viremic individuals, or the aviremic HIV-treated individuals. When looking at the PK, comparing individuals who had the 3 mg/kg on the right, or only the trace, we can see that the very consistent elimination when done with reactive counts, and also then compared the concentrations in the higher dosing cohort with reactive counts, suggesting that we did have good maintenance of the isotope with the antibody. So in terms of discussion, we did establish methods and a large collaboration to screen individuals into these sorts of trials and perform these complex studies safely. But ultimately, there was not any difference that we could identify in qualitative analysis of the scans between the 3 groups. Multiple differences between the human macaque model, probably importantly, the increased expression of target envelope in a macaque versus a human model, and the ability in a macaque model to give high doses of radioactivity per weight compared to the human model. The future studies, obviously, Tim Henrik will be presenting about zirconium-89 with that longer half-life radioisotope, and the increased dose of radiation that we can get with this isotope will now be more feasible in our setting with our study that we've completed. There's other strategies, potentially using a latency reversal agent to increase HIV protein expression, combining different antibodies, antibody fragment traces, or looking at actually targets of the immune system to image. A huge collaboration, huge thanks to everyone on this slide to bring many groups together to complete this work, but most importantly, the participants, and thanks again for the opportunity to present.
Okay, terrific. So James is not on the call today, but thank you, James, for that presentation. So we'll move on to our next presenter, who is Zaza Ndlovu from the University of KwaZulu in Natal in South Africa. And our second speaker after that will be Claire Delage from the Frederick National Laboratories for Cancer Research. Both of these presenters will present on flash talks for imaging. So Zaza, we'll turn it over to you. Thank you.
Thank you. Can you hear me?
We can hear you in this, and we can see your slides.
Okay, perfect. So yeah, today I'm going to talk about the work that we've been doing on imaging the HIV reservoir. My lab is based at the Africa Health Research Institute here in Durban, South Africa. We are interested in understanding the mechanism of HIV persistence in tissues. We also study CD8 T-cell mediated mechanisms of reservoir control or elimination. So we are very fortunate here in South Africa that we have access to a number of tissue samples. Have you heard from the previous speakers that understanding HIV reservoirs in tissues is very, very critical because most of the reservoir reside in tissues. We do get tissues from different sources, such as excessive tissue from surgeries. We also obtain tissues from volunteers that donate tissues, particularly lymph nodes for research and tonsils and gut biopsies. And we recently started obtaining post-mortem samples for research. And most of the work that we do is really focused on imaging, and we image HIV-infected cells and looking at HIV persistence in tissues. We also are interested in understanding the immune microenvironment around these reservoirs.
At the same time, traditionally, we've also done omics, where we disintegrate these tissues to study individual cells in terms of gene expression, in terms of function, in terms of immune function or immune dysfunction. Fortunately, more recently, through a grant from the Gates Foundation, my lab obtained a special geomics platform from NanoString known as GeoMax digital profiler that can actually combine these two technologies that we've traditionally applied separately, meaning that the tissue imaging as well as omics, so that we can really understand the cells in their native environment in terms of their function, in terms of relationship with the virus, as well as the gene expression. Most of the work that we do, the samples come from a study we call FRESH. I don't know if people know about it, but essentially this is a very innovative cohort where we recruit young women who are very high risk for HIV acquisition, and we recruit them when they are HIV uninfected, and we provide them with HIV prevention tools such as condoms and PrEP and other HIV prevention strategies, including economic empowerment. At the same time, we follow them regularly for HIV acquisition, and some of the women become infected, and when they do, they are placed immediately on therapy. So this has given us opportunities to try and understand the impact of very early antiretroviral therapy on reservoir composition, reservoir maintenance, and persistence. And so I'm just giving you examples of some of the women that are in the study do volunteer to donate an excisional lymph node. And when we get these lymph nodes, and in most instances, maybe after a year on suppressive therapy, 2 years, 3 years on therapy, in most instances, we do actually detect HIV antigens in the form of HIV GAG-P24 and HIV RNA in terms of GAG-PORNA. Here's just an example of one tissue, lymph node tissue, where we image for HIV P24 in somebody that was on therapy for more than 450 days. And we were readily able to detect HIV GAG-P24. And when we look into this P24 localization, we find that it's mostly localized in this BCL positive area, which is the germinal center. So more recently, we've been asking very similar questions to others: is which cells are infected and where do they reside? So trying to really resolve that. And mostly what we find in these tissues is that the cells of most of the cells that are actually infected or express HIV GAG-P24 are germinal center T follicular helper cells that are defined as CD4 positive, PD1 high, and CXCR5 high cells. And this we've published in the paper recently, but I'm just giving examples here of the germinal center T follicular helper cells that are GAG-P24 positive. We can process these to really understand exactly where these cells are and to actually enumerate these cells. And you can see here using histocytometry, where we can identify and enumerate individual cells. Here in this image, you can see the CD4 T cells are represented by green, the P24 positive cells in yellow, and the CD4 positive P24 double positive cells in blue. And clearly you can see, even without quantifying, that most of this P24 positive CD4 T cells are within this particular area, and very few are outside the germinal center. So we can quantify that, and you can see here when we do the quantification, looking at the distance from the cells in relation to the distance from the center of the germinal center, and also the MFI of P24 expression. You can find that most of the P24 positive CD4 T cells are within the germinal center, as well as the CD4 T cells outside germinal centers are P24 negative, and this is just a quantification demonstrating the spatial, localization of HIV-infected CD4 T cells that persist. One might argue that the P24 in the germinal centers that we see is residual P24 that is residing on follicular dendritic cells, which to an extent, most of it is, but there is indeed a lot of P24 that is on HIV HIV-infected CD4 T cells. And we also do RNA scope besides the P24. And here I'm just showing you two serial sections. One is stained with a GAG P24, the other one with we stained with we did RNA scope to stain for HIV GAG4. And you can see this mirror image whereby where we see the P24 is exactly the same regions where we're seeing RNA, suggesting that there's ongoing transcription in these tissues. And you can see here just a summary slide of very strong correlation between P24 areas and GAG P24 density. One of the interesting observations we've noted in these tissues is this heterogeneity in the different germinal centers. Here, the germinal centers are stained in green and P24 in orange. That in some germinal centers, you see a lot of P24 staining, whereas in some germinal centers, there's less P24 staining, and other germinal centers, we don't see much of P24 staining. So I've been interested in understanding the immune microenvironment around this persisting HIV RNA or HIV P24, whether that's different or HIV itself does change that immune microenvironment. So we've taken advantage of the new technology of or the spatial geomics to try and really understand the gene expression and cellular composition around these infected germinal centers. And so here we sampled 4 germinal centers. This is from the same tissue that has P24 staining, and 2 germinal centers that did not have P24 staining. And we applied this geomics workflow pipeline using an encounter readout that reads out about 84 or so genes that are pre-selected from this particular assay. And you can see here that what was striking for us is that in these germinal centers that had P24, most of the genes were downregulated. As you can see here, that there's less expression. Blue is less expression and red is highest expression. There were 2 genes that were particularly upregulated in the germinal centers with P24, which was FOXP3 and TBET. Whereas in the germinal centers without P24, there was a lot of genes that were upregulated, and FOXP3 and TBET were low, suggesting that Treg infiltration might be playing a role here. And indeed, we did staining for FOXP3 positive T cells in these germinal centers. And in the germinal center with the GAG P24, we saw infiltration of these FOXP3 positive CD4 T cells, which we did not see much of that in the HIV uninfected germinal center. So, so far what I've shown is that HIV infection persists in lymph node germinal centers for years, despite very early antiretroviral therapy. And it's very, very confined within the germinal center with fewer cells outside there, both in terms of RNA expression and as well as GAG P24 expression. And persistent HIV infection sort of induces an immune regulatory microenvironment. And these are preliminary data, and we're following up on these investigations. I'll end here and thank the people in my lab, the funders, the collaborators, and the study participants, and I'm happy to take questions. Thank you.
Beautiful work, Sasa. Thank you for that presentation. So we will have Claire give her presentation next, and then we will open it up for a Q&A for all of our speakers today. So Claire, I turn it over to you.
Thank you. All right, I'm gonna try to not screw that one up. Oh, of course. Here we go. Can somebody just confirm that you can hear me and see my slide and presentation mode?
Looks good, and we can hear you.
Thank you so much, Bunny. Okay, so thanks for giving me the opportunity to present a little bit of my point of view on the non-invasive measurement of HIV reservoir via imaging approaches. Giving a talk on imaging tissue with non-invasive techniques is quite of a challenge. So we all know that the issue, reside in the tissues, since the reservoir are within the many tissues across the body and are the main barrier to a cure. I stole this little presentation, this little figure from Kathleen and Jake review that was published in Current Opinion on HIV and AIDS in 2021. I highly recommend. That's a great read. And I represent all the different principal source of tissue reservoirs, which are mainly lymphoid tissues, where most of the target cells reside. So that's not a big surprise here. But let's be honest, at this stage, we all know that if you dig a little deeper in any of the tissue, you will find at least one or a couple of T cells or myeloid cells that are harboring viral, DNA or viral RNA. So right now, the best way to localize, identify, and quantify really the reservoir within tissue via imaging is to perform a lymph node or GALT or any tissue biopsies, right? And then you proceed to in situ analysis of the sample of a 5-micron section, right, of the tissue. And then you will look for cells, I will say, quote unquote, latent, who just harboring the viral DNA, and then some that are transcription active that will express RNA and DNA, and then the ones that are actively in a translation also. So you will have actually the full component of the virus, viral DNA, viral RNA, and viral protein. Obviously you can perform RNA scope, DNA scope, and combine this with immunohistochemistry and combine it to immunofluorescence and know a little bit more about those cells, the different type of cells that are harboring viral component, and so learn about the cell itself, its phenotype, and what other markers it can express. But we can now go really even deeper in the phenotyping of those cells using highly multiplex tissue analysis approaches. So because of time restriction, I will only present briefly one of this high-plex approach. This is the PhenoCycler Fusion, aka the Codex platform. It was developed by Akoya, and it uses a really unique technology of barcoded antibody that really allows you now to stain for up to 100 markers in one single tissue section. So you can obtain really beautiful pictures like this one. This is a chronically infected monkey, a lymph node monkey, farmer monkey. So, but here I'm just only plotted like 7 markers, and we stain for 37 on this one. And you can see that it's already extremely difficult for the eyes to visualize or to interpret any of those data. So the real appeal and the real power of those approaches is the spatial analysis capabilities that really comes, with the system. The fusion will map millions of cells in less than 10 minutes and can help us to really identify new population of cells and learn about the microenvironment of the tissue. So those are extremely powerful tools. Now we can detect viral RNA, viral protein, and any up to 100 markers to identify where they are and quantify them and characterize them. However, all those clever approaches are requiring a biopsy, which, let's be honest, is quite invasive to me. So it did not really enter the category of non-invasive approaches. I think the only approaches that are truly non-invasive, allowing visualization of the virus, are all the full body PET scan. I think James and Tim did a way better job and are much more better people to talk about that than me. However, I think there is limitation in the sensitivity of those approaches. Don't want to destroy the beautiful work that's being done, but here we're talking about visualizing virus cells that are already replicating virus. We're not really at the origin of the rebound, and it can be quite challenging to do those assays. So, however, I think there is one thing that non-invasive imaging approach can help us evaluate is actually the pathology associated with HIV infection. So I'm really sorry, but I'm gonna, for the rest of this presentation, I'm gonna leave the reservoir behind, the virus behind, I will say, and I will try to convince you that MRI can be a really useful tool to image the impact of the infection, which can help actually in the cure strategy mission. So we all know that the initiation and maintenance of effective immune response depends really on the structural and the functional integrity of the lymph node. And when HIV or HIV infection occurs, the response to the virus affects every lymphoid tissues within the body, beginning rapidly after transmission. And we know that lymphoid tissues are highly damaged during this infection, like enlargement of the lymph node, particularly the B-cell follicle. There is fibrosis that has been reported and so on. And we think that the associated pathology can impair the lymph node function, resulting in impaired immune response. So to evaluate the functional consequences, of progressive damage of lymph node, lymphoid tissues in general, we wonder if it was possible to adapt a non-invasive imaging approach to measure the lymph node integrity. So for that, we used a rhesus monkey model. I'm not going to go in details of this study design, but if you have any questions, should be in there. We injected a dendrimer in the foot pad of the animals, and we imaged the inguinal lymph node, and we saved the axillary lymph node to do parallel IHC analysis. So those animals were, as expected, highly infected. They were put on our 36 weeks post-infection and were also given an antifibrotic over time. 2 of them reached fully suppression at the end of the study. And look at the MX1 in the lymph node, we can see that there is obviously a high activation and inflammation in the sites. So here's a rapid, really 3D overview of the MRI results in a healthy, non-infected macaque, demonstrating that we saw following the foot pad injection of the dendrimer. We're focusing here on the inguinal lymph node region, highlighted in the cartoon here on the left. And rapidly after injection, we could see the contrast agent reaching and filling up all the lymph node in the chain, in the inguinal lymphatic chain. So, oops, sorry. Oh, there was a cartoon here. There was a animation. So we then conducted serial dynamic MRI scans over time following SIV infection. The slides show representative coronal section images to optimize visualization of the inguinal lymph node chain. So we have the picture that the monkey is laying down on his back here, and which is the back of the picture. The picture feet are down and the head is up, sorry. So on those particular pictures. So the longitudinal images demonstrate the change of dendrimer accumulation into the inguinal lymph node after a foot pad injection. We observe early and profound cumulative attenuation of the uptake of the dendrimer into the parenchyma, and the draining inguinal lymph node demonstrated by 2 weeks post-infection. And the attenuation of dendrimer uptake persisted despite the ART treatment started and the antifibrotic also. So we then measure basically the uptake in those inguinal lymph node. So you have the graph here, the percentage of dendrimer uptake relative to pre-infection. And you can see there is a drastic decrease of the uptake, suggesting a impaired of the function of the primary function of the lymph node, and that was not restored even with the treatment or the antifibrotic. So we showed that we could use MRI to follow uptake of the dendrimer within lymph node chains, and so in impairment of the dendrimer uptake as early as 8 weeks post-infection. We also noticed a stark difference in the pattern and distribution of the dendrimer in the lymph node of the course of infection. And you can see really that the dendrimer seems to go really around the lymph node and not within inside of the lymph node. So our first hypothesis was, well, this is it. The collagen deposition fibrosis is like impairing the draining function. But when we actually looked at the collagen 1 deposition in those tissues, we also looked at the collagen 4 this phenomenon really takes place over time, and it's a way longer pathology to come up. So the impairment that we see at 2 weeks cannot be explained by the fibrosis. So then we looked at simply our tissues, and we stained for different cell markers. And here you have the CD169, which is the macrophages staining. And I think it's pretty clear that pre-infection, those macrophages are more in the medullary core, T cell area, or B cell follicle. But here, post-infection, you really have those cells kind of taking over everywhere, but more particularly really under the subcapsular area. So we quantify that, and it was pretty clear that it is an enormous, I will say, recruitment, quote unquote, recruitment of those macrophages around the subcapsular area that could really explain what we're seeing in MRI. So to conclude, non-invasive MRI can really provide a longitudinal information on the lymph node function based on the dendrimer draining and uptake. HIV infection is followed by a rapid lymph node dysfunction manifested by a restricted uptake of the dendrimer by 2 weeks post-infection. And we think that the accumulation of the macrophages in the subcapsular area is likely perturbing the draining function of the lymph node. And ART or any other intervention did not repair this damage in the lymph node function. So we really think that, I think at least that, those non-invasive imaging approaches can actually help us to understand the pathology or the effect of a treatment, and can be extremely helpful for the cure research, not only just looking at the virus. So this is it.
Hi, terrific. Thank you, Claire. Very fascinating work. And thank you to all of the presenters today. Just really interesting technologies that you're using, and allowing us to look both deeply into tissues at the sort of with a spatial lens at viral persistence, but also in a very non-invasive way. So we can open this up now for about 15 minutes of Q&A, and maybe I will kick off the first question. And this is over to you, Tim. So one of the applications that you mentioned for the PET imaging was really to look at latency reversal or viral reactivation. And so I'm kind of curious, given what we know with current LARs that we have, where we've only seen marginal increases in cell-associated RNA, how much or how robust does the reactivation need to be before you think you would start to detect a signal? It's kind of tied to Jonathan's question that he had in the chat about, when do you decide in an ATI that you're going to start to image, and are there any correlations with PET activity and the timing of viral rebound?
Yeah, no, it's a great question, and that I think as we're trying to push our sensitivity a little bit higher with some of these novel techniques like Explorer imaging and getting the right antibody tracer combos together, it's probably we don't know, I guess, is the main question. But I think that's something that clearly is the next step that we have to look at. What is the threshold and the sensitivity to look at changes dynamic when you look at this? And I think that with our ATI studies, we're now going to move towards imaging much earlier after removal of ART. So probably before, remove ART, and then basically look very carefully within the first week or so, long before we start to see reactivation, to see what types of tissue changes we can see in dynamics. Because we obviously know that there's going to be fulminant infection in those tissues long before we see a breakout, based on some immune sensing from Lady Troutman's work and others. And so I think that that's going to be a key question, although I think we will be able to see something. I think that looking at the TGF-beta paper in SIV, for example, was fascinating to see that, they were able to see such a dramatic response in reversal of latency in treated animals using immunopet imaging. And so I think that's going to be key. There's a lot of challenges with humans in terms of the sequence and the avidity, binding. You don't necessarily need neutralization, but what's the binding of each of these antibodies to the patient. So it's going to be on a patient or a person-to-person difference as well when you're doing some imaging. So, but I think we're going to have to look at that and do direct tissue correlates as well during the same time to make sure that we know what we're looking at is real, and we can actually quantify using things like a geomics or high multi-parameter tissue-based imaging, which we're doing in combination with our PET imaging now as well, to really see what's happening in terms of the sensitivity of finding whether it's HIV proteins or transcriptional activity, et cetera. Lots of unknowns.
Exciting work. Thank you. I see Deborah has her hand up.
Yes, hi. Thanks, Bonnie and Tim. Terrific session. I actually, from James' presentation, it occurred to me, and maybe this is obvious, that knowing baseline susceptibilities to these BNAbs are going to be critical for their use and application for imaging studies. And I wondered what the feasibility, in our base, in our recent clinical trials in children, and I hate to bring the word children into this space, but they are part of the cure agenda. About 27% of kids in sub-Saharan Africa had baseline resistance to VRC01. And so so it seems as if you will need pre-screening to be able to use these imaging technologies or combinations of antibodies to be able to assure their use. So is there a way to use a combination of these antibodies for the labeling studies, given that there's going to be baseline resistance?
Yeah, maybe, I don't know if James, it's probably 2 a.m. for James, so I don't see him on. But yeah, I think, yeah, you certainly can use a mixture of antibodies. So you could mix, you can't tell which antibody is going to bind because positrons are positrons, but you can certainly mix a combination to try to engage or increase the binding. I should mention, though, that you don't need full neutralization. So you could potentially have a scenario where you don't fully neutralize a virus, but you do have binding sufficient enough to image. And so I think we have to parse out a bit how much of the, how much it's really the avidity of the binding more than it is the neutralization capacity of that antibody. Now I know they go hand in hand often with this, but there might be a dichotomy there. But you certainly could use a combination. I think one of the challenges there too is that pre-screening in a suppressed participant for a long time is really hard. I mean, we've been working with Monogram to do the neutralization test. We've been trying to genotyping from DNA, which is not great, and it's hard. A lot of the times you just can't actually get a good screen from someone who's on a suppressive ART, and that's a challenge. So we've certainly played around the idea of combining different antibodies that bind different parts, whether it's a CD4 binding versus a V1, V5. To look at different ones and say, okay, we don't know which one is binding, but at least if we know that one of those will have some type of avidity. That said, it's going to be difficult to cross-compare, but at least we can look at changes and to do that in people. So, yeah.
Thank you, Tim. That's helpful.
Terrific. And I see Linda D has her hand up. Linda, do you have a question?
Yeah, I guess it's more of a comment than a question. It's been very interesting. Tim, for you and for all the assay people as well. I mean, I have a tendency to be very practical, and I'm just wondering, as far as your technology, Tim, how available is that to sites that are not involved? I guess you have to be involved with an institution, but as far as the assays as well, I mean, I just think that we need to have I know how complicated this is and how many things we need to consider, but to try and make this as easy as possible. I guess scalable is kind of not the right word, but you know what I mean when I say that, so that everybody will be able to reproduce this and conduct this and hopefully reproduce positive results. So I don't know. I just hope that people are thinking like that. I mean, I know sometimes it's easier. It's harder to even get to that point because you have so many scientific things that you have to be concerned with. So anyway, just a plug for keep it simple, stupid.
Yeah, no, super important. And by the way, with the PET imaging, all the center needs is a PET MR or a PET CT scanner that has a reasonable level of detection. The zirconium labeled antibodies that we're making, for example, we could make locally and mail across the continental US or even abroad. We actually make the antibodies a day before we actually infuse them, and then we image 3 days after that when we start. And so these things are long-acting isotopes. So, we can actually create it, locally, or another group that has a large nuclear medicine program, and then basically ship it to centers that just have a clinical PET machine. And you could actually do this across platforms. So it's expensive. I'm not going to deny that, but it certainly could be, outside of centers with large nuclear medicine centers, but at least that have PET CT or PET MR technologies available for diagnostics.
Okay, cool. Thanks.
Okay, terrific. I don't see any other questions in the chat. Deborah, did you have another question? Your hand is still.
Yeah, I just had, one more, if there's time for one more question. So, and this is for Tim. I know, within the pediatric space, we're developing the AAV vector delivery in the non-human primate for long-term production of BNAbs, combination BNAbs, and the consideration about using these antibodies for imaging has come up, and to what extent this would interfere with the outcome or, competition for the binding sites. And I wonder your thoughts on that, because I would imagine some of these ATI studies will be in the context of BNAbs in humans. So what are your considerations around using labeled BNAbs when we're trying to assess ATI effects with BNAbs?
Yeah, it's a challenge because the amount of BNAb that we give for imaging is minuscule. It's a microdose. It's like 1/100th or less of a mass dose of what you would give for treatment. And so the problem is if someone is on BNAbs, as part, we had this with our AMPHAR study trial that there's a combination of VRC07LS and another non-competing antibody in the 1074. And so if you have the VRC07 also competing for the CD4 binding site, then you could potentially completely mask that immunopet signal from the target. So you either need to have a monoclonal that has a different binding site than what you're using for treatment therapeutics, or you'd have to find other another modality to do that. So in that study where there were BNAbs, we actually been using our T-cell imaging technique, leveraging our DARE establishment to kind of indirectly look at kind of immune response within that setting. And so that is going to be a challenge. But this AAV delivered, endogenous production of antibodies is fascinating. I think it's amazing. It's a great, kind of strategy going forward therapeutically. And so it would be great to have some type of technology to be able to parse that out and to look at differences. But yeah, that is a challenge when you have BNAbs that compete for the same binding site.
Thank you.
And I have one last question for Claire. So beautiful work again. I was fascinated by the PhenoCycler platform and the ability to look at 100 markers simultaneously. And just curious from a technical standpoint, are these panels that you have to put together and validate the antibodies? And then more importantly, how do you extract data from 100 different markers in a meaningful way? And how long does that take?
Thanks, Bonnie. I think this is a really important question and point to make. So the Akoya platform, the way that it works, like you can basically conjugate your own antibodies. So which was a big plus for us because this technology comes from like the cancer research and was performed a lot in human and cancer markers. So not really for HIV or SIV purpose. So we were able to actually use the antibodies that we developed and validated in the monkey model and human model and human tissues and conjugate them ourselves with the barcode. So I will say that is the easy part. And the technique itself is also actually the easy part, believe it or not. So it takes, 2 days, really, not really even 2 full days to like run the full thing. Where it gets tricky, this is where you want to analyze your data. So yes, there is many. So Akoya provides a really remarkable software to study, neighborhood cells and identify cluster and population of cells. So that is, I will say, also again the easy part. Where it gets a little bit complicated, this is where you want to have something really analyze every single of whatever number of markers you put on your slide. And this is where I always come with what's important, and it's gonna join a little bit of what Nicola was saying. Like it depends on your question. So at this stage, because the analysis is so time consuming, we have to come up with more precise, narrow answers and look for that in a time manner that will be correct, I will say, and we will, answer specific questions, but taking really just the full possibility and the full data that we can get out of those 100 markers is really endless, and there's a lot of things that we actually don't even know yet. So yeah, it will take months really or even years to analyze all that. So I will say, let's stay precise and question-oriented hypothesis.
All right, good, exciting potential there. Okay, I saw that Tim's hand was up, but I think, Roger, are we going to move into the breakout sessions, and then maybe Tim, we can get back to your question, gather again.
So thank you very much to all the presenters. We have a series of very rich and intense presentation of technologies, and I can see there was a lot of questions. We're going to move now into breakout room. We're going to split participants in two. We have allocated you to a room to make sure we have a mix of people, and that's important. We really want to hear from everybody here. We have a range of people. And in the blue room, Bonnie will lead the conversation, and in the red room, it will be Tim. We have already put a number of questions on top. You can address some of these questions. If you have other questions, it is fine. What we're looking for is really a lively conversation. We will be in the room for about 25 minutes maximum. I may move people from the room if I see a huge imbalance in the number. Tully in the red room and myself in the blue room will put some post-it on the screen to keep the conversation going and to allow for a summary at the end. I will also share the link to the board for those who prefer to post their comment themselves using post-it. Just make sure you're posting them in the right room. You got the name of the people. This is done here. And that's all for me. I'm going to let my colleague at IAS trigger the breakout room. People are rejoining and taking over Tully while she's closing the other room. Oh, here you are, Tully. And I'm sharing my screen for the reporter summary, which will be brief. And this will be followed by the final discussion on everything that will be said. So we're nearly there, but I see there were more questions. So I'm just going to stop talking and hand over to Bonnie for a quick summary, then Tim for a quick summary, then the final Q&A.
Okay, all right. So just to hit on a few discussion points from our break room, we did discuss whether or not you have to worry about the defective genome, recognizing that defective genomes can contribute to some protein production, which can influence immune function and dysfunction and inflammation. But I think consensus in the room was really, if you're focused on cure, then focusing on the intact genomes is probably the most relevant measure to make. However, we did recognize that if you are working with interventions that are potentially targeting proteins, and that in these cases it might be really important to also look at the translational component or the protein components of the reservoir that will allow you to better assess the effect of your intervention. And I think Matthias showed a nice example, or talked about a nice example around shock and kill and Nicola as well, and that sometimes you can see with your interventions effects on both the intact and the defective genome. So that was something to consider. We actually started the session by defining non-invasive assays and markers. And so while, PET imaging, MRI are certainly quite non-invasive based approaches, the other means of collecting blood, urine, stool are also pretty non-invasive ways of collecting samples. So we sort of felt that they all fell within this category or topic of non-invasive assays. And certainly the tissue collections are the more invasive. So not really a big challenge to be able to do things when you're sampling blood, urine, or stool. The question was asked, can industry help bringing assays to scale? So we had a discussion around that, and there's certainly opportunities there, and an example was given. We talked a little bit about the application of these imaging approaches, especially PET-based tools, which are, showing some interesting data, but clearly have some, ways to go to be able to understand how best to leverage these and in what research settings. There was a question, and so maybe we can talk about this when we have our group Q&A around how well those, how sensitive those approaches are, and it may be dependent on the antibody that you're using, but can you see CNS, can you see brain reservoirs, and how do you really distinguish the signal that that is coming up between the different compartments? What else? I think that might hit some of the highlights. Is there anything else from the Blue Room group that you want to mention here that I didn't cover? Oh, I think maybe there was one other point, Matthias. We talked about this in the end, and there was just a discussion around the importance of figuring out the biomarkers to understand people who are not going to rebound. So we've been focused a little bit on that time to rebound, but also understanding who's not going to rebound and distinguishing responders and non-responders sort of retrospectively by making various measurements in your studies. And then, after some level of validation, perhaps you could use that more when you think about enrolling individuals into clinical studies. So I think I will end there, maybe turn it over to Tim, and then we can come back and talk about any of these topics in our group Q&A.
Oh, great. Thanks, Bonnie. Yeah, I think we actually had a lot of overlap, would be my guess, as expected. So I'll try to keep it brief as well. But, I think, certain things that you can see up here that we're talking about need to figure out best ways to kind of integrate lower technology, kind of lower tech, but scalable to reach the relevant patient populations in the right context and the geography. And, and obviously also need markers that are, something that can be easily reachable cross-platform validation, but also looking at the macro environment, what types of markers in the setting of API, and expanding studies on ART to look at kind of this residual and how these correlate in the setting of suppressive ART. I think, but we also came down to a lot that tissue, as I think Helen summarized, is the issue. This is a tissue-based disease. And I think we need to think about prioritization in terms of what studies that we want to validate cross-platform in these clinical trials that are going to get not just at pathogenesis, but at a meaningful outcome that we can actually measure, or a biomarker of potential success, which is, I think, the key problem here is that unlike a viral load, I'm ad-libbing right now from our group, but unlike viral load, where we have, clearly it was clearly related to increase in CD4 T cell counts and increased mortality when you reduce, we don't have that yet for reservoir measurements, right? And so, is it going to be in the circulation? Are we going to be looking at metabolomics that then have to be cross-validated across different, different platforms? But I think the one thing that we really want to get down to is how do we do this with resources? And these are expensive studies, and really how can we work with industry to really motivate, either through foundation type structures, initiatives, or philanthropic parts to support these types of assays that may not be viable commercially in terms of diagnostic platforms for a larger rollout in clinical therapeutics. At that point. And so I think that's the real challenge that we thought, and kind of where we need to kind of work forward, is to really figure out how we can partner academics and industry to really, get these platforms across different studies, validated across different studies, and implemented in basically all regions where we need to be doing these types of studies. And maybe I'll just sum up with that and see if anybody else in the group had anything else. Many things I missed, but just to keep it short so we can reach the discussion.
Thank you. So I think I'm going to hand over to who is leading the final discussion, final group discussion. I'm going to still keep some post-it. I can also repost some of the things that I had put on top as a memoir if you want to carry on that, or if you want to carry on with questions you didn't have the time to ask in your group discussion. I'm handing over to Tim and Bonnie to lead together this joint discussion.
Yeah, thank you, Roger. So maybe I'll just pick up on the last remark that Tim had captured, which was around can industry partner, and I definitely do think that there is an opportunity there. Obviously speaking on behalf of my experience at Merck, and we've been very much committed to leveraging some of the technologies that we may have access to or the capabilities that we have access to and figure out how best to partner. And oftentimes it is sometimes in these cutting edge technologies, sometimes it's kind of applying things in other therapeutic areas. So I think Mohammed mentioned this as well. There are, we're learning of how we detect the rare events of the cancer cell in circulation, early on or even before treatment. So how can we leverage like technologies or like approaches? And so from biomarker discovery to assay development to implementation, we do try to invest where we think that we are best situated to make that investment. And of course, there are lots of other types of industry partners, and I think we should find ways to engage them. But I was describing the work that we're doing on the TASO in-home blood collection for viral load measurements. And Linda and Jeff are on the call. They've been really helpful in working with the community and our study participants to understand the level of engagement, to be involved in studies that are maybe exploring new assays or new detection approaches. And so we talked about that. But then we also, I sort of mentioned how we were planning to use another partner, if you will, an industry partner, to automate that whole workflow of taking the sample off of the device and making the viral load quantitation. And it was becoming more challenging to do that. So we internalized the whole thing and we built a fully automated assay. So it goes from extraction off of the device to data out on the back end. And we're really, happy that we did this and looking forward to the final results of the study. But we will make all of those methods publicly available. I've already reached out to some other assay companies, if you will, to ask them if they would be interested in, adopting these methods to be able to enable this at a larger scale than we could do sort of as a single company. So I don't know if there's any other sort of thoughts around how best to engage in industry academia partnership, but if others have ideas or asks or a perspective on that, feel free to chime in.
So Bonnie, let me ask you, it's Linda. I haven't focused a whole lot on assays and assay development, but has there ever been a big meeting to discuss this, like we did for the ATIs at Raygon, for instance, where people could come together and talk about this exactly like we're doing today? Maybe now that we can get together personally, speak together in the hallway and forge some sort of collaboration, how things used to work.
Yeah, not at that scale. And I think it's a great suggestion. There are interactions through the Martin Delaney collaboratories. So I think we get we have more cross-fertilization in that forum. But a meeting like that would be a great idea. We can invite many more potential partners and stakeholders and really figure out how to tackle this.
Yeah, because, initially when industry collaborated 100 years ago on because they knew essentially that it was going to take combinations to be effective. I mean, I'm not sure that the assay companies have that same sort of promise at the end of doing this. So working together with a Merck or a Gilead might be helpful, but I don't know. In other words, I think that getting together seems like a great idea, and I'll save my community comments for the end.
Okay, great.
Thanks.
Omar, you had your hand up. It is now down. Did you have a comment or?
Yeah, I had a comment, and my comment was around community engagement and social behavioral research. And I was wondering if there is, if anyone has information around acceptability of these techniques by the target communities, especially the vulnerable communities that are high risk for HIV. I'm just wondering if we are discussing techniques and assays that will be acceptable to the target communities.
I think it's a really excellent point that you raise, and there's probably multiple levels of it. It probably depends in part, what the ask is or what the assay is or how invasive or non-invasive the sampling is. So, this is where we, I think in general, just need a better perspective from the community about what is acceptable. I know when we were gearing up to do this in-home blood collection device, and this was asked in the break room, is whether or not there was enthusiasm or acceptance for this based approach. And in our case, there was quite a bit of interest, and we were very fortunate to partner with the community to be able to enroll participants in the study and also have surveys sort of broadly sent out to understand the community perspective of this and, what would be the opportunity around it? What would be some of the challenges? So I think that's an important component that we have to build into our thinking when we develop these and try to apply them. But we would love to hear your opinion as well, and others.
I'll chime in again. Jeff, was that you?
No, it was Carrie and Laura.
I was going to kind of save this for the end, but, I think it's really important that when you talk about how you're going to enroll participants for these kind of studies, you're probably going to be talking about 3 and 4 hour procedures as opposed to blood, a simple blood draw. So, I think it's really important to realize, everybody makes money in this game except the participants, I think it's really important that they be compensated fairly. Without the ethicists coming in and saying, well, this is really, past an overly, I don't know, you know what I'm trying to say, incentivized that so the patients can't really decide without saying, oh, wow, I really need this money. I better do this no matter what. But there's a way, there should be a way that we can look at the levels of reimbursement at this point. I don't think they've been raised for a lot of things for many years. So, it might be time to look at reimbursement levels for something like this, where there's not even a hope of any sort of benefit for the patient for sitting there for hours and hours with this kind of thing, tested and necessary.
Yeah, Linda, this is Jeff, and I'm glad you raised that because several of us are working on that. Corinne Dubé is part of a team with Brandon Brown at UC Riverside, looking at exactly that in the context of peer research. And you're right, it's the emphasis has always been about avoiding coercion as opposed to fairly compensating people for what they're providing. So we really need to look at that. So there's a lot of research that needs to be done. And I keep saying it over and over again, but we need more socio-behavioral research, and Corinne Dubé is the one leading the charge on that. So we need to find a way that we do this across networks, and there's been talk of creating a core that works across networks because the ACTG, MDC, various places are doing this. And yeah, it's hugely important as we move into clinical trials. Thanks.
Terrific, thank you. And I see Alan has his hand up.
Yeah, thanks, Bonnie. So I wanted to follow up on the development of the assays because we have on this committee done in the past, talked with the diagnostic industry, but what we find is it becomes a heavy lift for developing assays. And at the end of the day, unless there's some value to it, hasn't moved forward. And I think in our session with Tim, we talked about how do we get, maybe some of these more philanthropic arms of the industry side. I mean, that could both be diagnostics and pharmaceutical groups to think about the really leading edge assays like imaging assays and a lot of the single cell assays that are still not ready for prime time, and how do we invest in evaluating them, and especially on a global basis. And I think, as was brought out by Linda, yeah, there are a lot of groups. Mike McCune has a meeting of his cohort of the groups doing the diagnostic work on the Cure in Africa group. So I know there's, they're having one at Gates, but that's separate than what goes on at the meeting in Miami where the Cure group comes together, which is going to be separate what the ACTG. I run a retreat for the lab group at the ACTG with the TSGs where we'll talk about this. So we're all a little bit siloed. And I think at the end of the day, we need to figure out how do we get, in this case, a global audience together, and maybe the IAS can help. Roger and Tuli can help us identify all these groups and then bring them together in a room. So we do have the communication, and especially with the community, as you, Jeff, and Linda have said, is the burden on the patient in doing these assays.
If I can comment on that, Bonnie. Yeah.
Yes, please.
So I also support the another group led by the IAS, which is called the Industry Liaison Forum, which some of you may know. And we have done some work specifically about diagnostics development and the challenge of developing diagnostics, which are much more complex than developing drugs, especially when you want to roll them out in low and middle income country. That's an ongoing work. We have been able to put a group of different stakeholders together. I have to say it is challenging because there are not that many assay developer on it, and it's complex and it's very costly compared to drugs. But I think it's that's why I wanted to, we are reaching the end, and I really want to ask that question in relation to this meeting today, which really could be the first in a series that, as you suggest, Alan, the IAS could dig deeper, and I hope that's going to happen, is how are we going to move forward with the non-invasive assays compared to the gold standard currently used? And why I'm asking that also, my field is HIV vaccine. And I know that for 40 years, people have been developing all kinds of assays. And at the end of the day, there is always this question of harmonization and what they mean, which assays to use. So we are quite in a younger field, in the cure field, and how people feel about what's the next step. We have about so many assays that, and I admit I don't know many of them. How are these different academic groups going to move them forward, ensure that the industry is on board because they also conduct research and studies, and that I would say, moving in a better way than the HIV vaccine field. I can say that, more coordinated, I would say. Challenging question, or nobody wants to take a risk, and it's really about coordinating.
I was going to say, I think getting the different stakeholders, you're right, there are so many different, and going back to what Alan was saying earlier, I mean, there's so many different kind of groups independently talking about this. Some people are similar in those groups, right? So there's a lot of crossover as well, but I think it's really how do we engage this kind of larger consensus? And I think it's hard to approach a company. It's hard to approach a philanthropic organization. It's hard to raise funds or to even get grants unless you have a unified consensus within that field of what you want to do. And that, it's like trying to ask for something that you don't know what you're doing. It's going to be difficult. And so that's going to be an absolute key part is to really harmonize and synergize. What does this field think should be the measures? Are they going to be cure strategy specific or prevention specific or treatment specific? And if so, what should we really be focusing on? Because I think if we have a unified voice of what these measures are, it's going to be a lot easier to leverage corporate support as well as other things. We've run into this with long COVID, right? And with SARS-CoV-2, my other full-time job now, right? I mean, we've been trying to, engage companies to provide drug and to provide various, different platforms, but it's very difficult because A, we don't know what causes it, and B, is it worth taking a risk for something that we don't know if there's no consensus? And I think if we really reach on that, then we'll have a little bit more leverage in terms of working together, if that makes sense. I know that's a little bit esoteric, but anyways.
So I guess I don't hear anybody talking. I'll just forget about raising.
Oh, Linda, go ahead.
It's okay. No, it's
Oh.
all right. I'm trying to be good because usually I never use this raise your hand, but I'm trying. Anyway, I wonder if it might be a good idea for the IAS industry group. It seems perfect for this to me. I mean, it needs to be sort of industry sponsored or involved in order to get the people that we need into the room. And it may be that the whole meeting wouldn't be this, just these sorts of assays. I mean, just the idea of that tacit device, I mean, that is so appealing to the community, and especially in this COVID era of not having to go to the doctor for blood work in a regular ART study. And really more importantly with ATIs, to be able to have a handle on what your viral load is doing during an ATI. But in other words, if we kind of had a meeting where we talked about both of those things generally in ARV studies and how much money there is to be made there because people are so, participants are so interested in this. And I really think they are. I mean, every time we talk to people, and I think that Jeff can chime in here too. I mean, I think it's like, wow, this would be great, So, maybe, I mean, and money is money and that's what it is. And maybe if we made it a more appetizing meeting as far as how are we going to make money here, we could slip this other in and, in the afternoon or something and see what we have and see if it grows. I mean, at this point, we have nothing that is coordinating all of these different entities, these different groups. So.
Yeah. And I guess thank you for that comment. I think I would just echo what I'm hearing others say is, bringing folks together, building consensus around what it is everyone needs and would be interested in having. And I can tell you that, if you partner, say with a company like Merck, if we partner with someone on an assay and we are going to help them in their space deploy it, and usually it means at scale, but we would like to then kick the tires on the assay and the studies that we're running. And in order to do that, we have to build it at scale. So, and then, I think we all benefit in the sense of we can then, sort of have something that's a little more high throughput and that can be shared across different companies and different, communities, potentially recognizing that there are challenges in different resource settings, but it could be a better starting point. And it really, from my perspective, it requires this dialogue. It's this understanding of what the needs are and what the asks are, and how do we best partner with this end goal of trying to deliver our cure, or just, in general, support HIV eradication efforts.
Yeah, I saw Jeff's comments in there too, and Jeff, we had this roundtable a few weeks ago looking at ATI monitoring in the setting, and I thought, yeah, a lot of crossover with this discussion as well, right? Like, how do we do this, in a point of care wet method? It's palatable and easy, and clearly it's connected with our whole, reservoir measurement and outcomes. Here's the thought, as I hear Matthias, talk about him Jews assays, and every time they publish it, it's like it adds another layer, right? So they can look at full length sequencing, they can look at full length sequencing integration site, they can look at full length integration site on a single cell level, and now with phenotypic, with antibody data, and now we can do this with various geomics and cosmics, DNA spatial profiling. What's interesting about this is these don't have to be HIV specific. These platforms have HIV specific features to them. Like, for example, if you're looking at an in-target RNA that's from HIV. But overall, those platforms are actually, they're adapted from general research, right? And these things could be, if we're looking at scale, we're looking at how to entice this to the corporate world in terms of, we're not going to just make this one very expensive platform for a single, niche market. And I don't mean niche market I mean it only in the setting of, looking at the global kind of finance and things like that. I don't mean to belittle any type of disease process that is studied, but if you could do this cross-platform and say, look, this is widely applicable to, for example, cancer therapeutics, right? I mean, cancer is huge, and you could adapt these types of things to look at circulating cells for all sorts of different malignancies that are up and coming. So if we could somehow kind of integrate some of these platforms, which are becoming more and more standardized, despite the fact they're more complex, right? As we're using these kind of standardized technologies that are becoming more readily available to the general lab. Those would be great. And so if we can, like, you know, piggyback onto cancer therapeutics or autoimmune disease and say, look, we're actually looking for similar metabolic profiling. These are non-viral markers. These things are also perturbed in many other disease processes. And so that would be one way to really kind of, you know, anyways, just a thought, because this doesn't need to just be HIV specific, although it can have an HIV specific part to it. The general platform could be much more adaptable. So I just wanted to throw that out.
But Tim, I think the point being, as you said, when we're thinking globally, we're thinking also non-communicable diseases. So when the World Health Organization looks at the biggest impact, it's all about the NCDs. It's cancer, it's heart disease, it's metabolic outcomes. So, I mean, the intersection here between infection and non-communicable diseases is huge. And Jeff just said it. What about Ebola, monkeypox, SARS, all of this point of care? So I think any infectious disease, malaria, TB, all of this is in the hopper to be looked at. And HIV could lead the way to help many of the other infectious disease diagnostics and pathogenesis questions. So I think, you know, and as we're thinking about preparing for another new pandemic, you know, disease X out there, what's that one going to look like? So I think it's all very good. And maybe, you know, again, the IAS could look at it more broadly about what's affecting the HIV population, because this is a population impacted by all of these global health issues.
Yep, fully agree. And the way I look at it is imagine and then apply and then reimagine and then apply. And then you just kind of get this sort of expanded scope of the, you know, those ideas. So, and it looks like Heather has her hand up.
Thanks. I know we're about at time, so I'll try to keep it short, but I work with Mike McEwen on the HIV Frontiers work at the foundation, and I love every part of this end discussion. My background is in oncology diagnostics. So that's the kind of world I came from in training those biomarkers. And we've been having a lot of these same discussions to, you know, the point earlier about these different siloed groups and how we come together. We've been having some of these same discussions on our end as well. And me really thinking a lot about how can we leverage the assays and the development work in the non-viral space that's already been done, and how does that kind of segue into the HIV reservoir work that's being done as well. And so excited to continue those conversations that very much in the same brainwave as a lot of what was just discussed at the end and how we engage.
So maybe the first session of our meeting should be about the intersection of these things and talking about cancer and other sorts of conditions right off the bat.
Yep, great idea. And thank you, Heather, for sharing that with us. So I think that brings us to the close of our session. So on behalf of Tim and myself, Roger, Tuli, the participants, the speakers, thank you everyone for joining us today. Tuli, did you want to make some closing remarks about the recordings and availability after the session?
Yes, I will happily make a couple of comments. Yes, indeed, we will be sharing the recording together with the report in a couple of weeks' time. So we will be reaching out to all the registrants and the participants. If you happen to have any questions, particularly about this meeting or anything that is in the pipeline for the Cure group, please do reach out to us. Yes, thank you again for joining us. For final words, I will hand over back to Bonnie and Timothy.
Okay. Tim, closing remarks?
I think we just summed it up. I think with our discussion. So no, thank you everybody for this. And it was really dynamic and fun. And hopefully we can really leverage this as with the ATI roundtable too, to really kind of get some movement on the ground and some traction to finally really push forward at getting this implemented on a larger scale. So that's our difficult next task. So thank you, everybody.
Thanks.
Yeah, this has been great. Thanks, everybody. Good job.
Thank you.
Bye, everyone. Thank you.
Bye now.